So getting started on our ATACseq course for today. What is ATACseq? Well, ATACseq is assay for transposable accessible chromatin using sequencing. So effectively, it's going to use a transposase to fragment accessible DNA. So regions we would consider more open, not so tightly compacted chromatin. So this should allow us, in combination with sequencing, to get this map of where our open chromatin is, where we have less compacted chromatin. So perhaps some activity there, transcriptional machinery, transcription factors, places of the genome we're interested in. There is epigenetic regulation perhaps occurring at these regions of open chromatin. So in contrast to other techniques, and I think why ATACseq became so popular, it really required a low input material, rapid, very easy to do, or rapid to do. I think it must be, well, I don't know, because I know it'll be in the lab, but it seems like people have really nailed this. It seems like we get great data coming from it. So it's become a really ubiquitous technique. And simple, as I said, we have this compacted chromatin and open chromatin. We use our TN5 transposase. It can go into where this isn't so compact, so where it can get access, and it will make cut sites, and we can pull down or we can collect all these fragmented bits of DNA. So where our transposases come in and cut our DNA, we can then take this DNA, right, it's fragmented, we amplify it and we sequence it, and then we get our representation of where this T5 transposase is able to cut. Hence, we get an idea about where is open. Right, so our other techniques do something similar to ATACseq. So famously, or a very popular technique was DNA-seq, very expensive technique too. And this was an enzymatic digestion of open chromatin, really around transcription factor binding sites. So it had to be very open chromatin, and really you're just capturing these transcription factor binding sites, which is actually what we want to get mostly from ATACseq. We then had ATACseq. So my wife really did a lot of ATACseq. This is enzymatic digestion around nucleosomes. Okay, so actually this requires a lot more sequencing, and we're going to get all the nucleosome positions. But ATACseq allows us to get a little bit of both, right? It uses the transposase and it offers us a method to hopefully extract signal around transcription factors like this, but also around nucleosomes, which are in a little bit more of an open chromatin. Okay, so we're not going to get the nucleosomes from our highly compacted chromatin. Our TN5 transposase won't get in, but it will get in around areas where you have nucleosomes with some free chromatin around them, some open chromatin around them. Okay, so we tend to see something like this around our open regions. We'll see for ATACseq a lot of signal around our transcription factor binding sites where we have these open regions in our genome, and then less kind of tailing off. So in this session, we're going to take advantage of lots of pre-processed data, and because ATACseq has become so popular, there is so much data out there we can really take advantage of. And we're going to look, I think in this session, not post-alignment so much, but we'll look at a bit of QC, and we'll look at how we can do alignment. Over the process of these sessions, we will get through alignment, post-alignment, some plotting of the ATACseq signal, as well as, in the end, some motifs, right? I think the entire last session is on motifs. So because this is, I originally did this as a workshop, actually we have more data prepared for this than I think we do any other course. So I went through and I actually make use of 3 published data sets. I went and got the first paper for ATACseq. I think this is the first paper. So by Buenrostro and Greenleaf. So we have this process somewhere. We also have, you can go get the actual raw data here from GEO. The second data set we're going to take is from ENCODE, and the nice thing about ENCODE is they had some replicates, whereas this first data set I don't think we did. So we're also going to make use of some liver, kidney, and hindbrain data, where we have, I think, 2 replicates, and we'll try and pull out changes in open chromatin DNA accessibility between these. And finally, I've taken some data from Christina Leslie's lab across the road. No reason why I'm picking on Christina Leslie. I just saw that she had some ENCODE data, which was a little bit more recent. So I wanted to take that because this is quite a new technique, newish technique, and it's got a lot better over time. So if we were to compare this first paper, which I haven't got the date, I should have put the date here, versus what was coming out of Christina Leslie's lab for ENCODE, the difference in quality is dramatic, but that really should be expected. This is the development paper. This is practical use for ENCODE. So you can get the FASTQs here. You can also get the BAM from here. Going to need some reference data too for this. We have our reference genome in FASTA format. We know we can get this, and we've been doing this in the bioconductor, but also in ChIP-seq yesterday, and in RNA-seq perhaps on Monday. So we can retrieve all the FASTA format, the whole reference genome, everything we need to align to from our BS genome objects. We have gene models, so not necessarily, for RNA-seq, typically we would definitely need gene models, right? We want to summarize over genes. For things like ATACseq, ChIP-seq, we need gene models because we want to associate this open region, this where we suspect a transcription factor is binding, is next to this gene. So we need to know where genes are at the very least. Blacklists, so we also, I think Doug talked about this yesterday, and we have this nice cartoon showing the effect of blacklists. There are artifact regions in genomes, so perhaps regions we're not fully sure of the sequence, or we don't know that there are other places in the genome. We suspect there are other places in the genome which have very similar sequences. These tend to accumulate lots of signal. This is an artifact, and the blacklists are well known, and actually you can find maps of them on ENCODE, and these are areas you might want to ignore. Okay, they're probably useful information there, but because they have such high signal, because we're not really confident in that regions, you would need to do very different analysis to investigate them. So we're going to start with some public sequencing data. We're going to take the links below and use this reference data we just mentioned. Since some of the processing steps may take a while, like I was saying, I have pre-processed some of these for you at these locations. Is this me? This might be the ENCODE link. Here we have. We also have peak calls, some processing of this full data available in this little download here. Okay, so it tells you can unzip it. So if you wanted to follow along, we have some processed for you data here. Same as we have here, we also have the full data. So this is really the essential. We also have the full data. So this includes BAMs and other aspects. We also have bigwigs. So these can take a little bit of time to create. So if you wanted to and see the effects of what we're actually doing in the course, you can just take our bigwigs from here, or you can generate them as we go along. So let's get started. So the first thing we want to need to do really with any data, we may not do anything with this information, but we are going to do some FASTQ quality control. Okay, we need to know, does our data stick out by metrics which we've reviewed? Is it different from all the other samples we're running at the same time? Is it different from what my genome center will typically produce for this type of data? Is it different from my friend who just ran an ATACseq last week? His data looks great, mine looks rubbish. Let's compare our FASTQ quality to see if I can identify anything. Okay, so we always want to recommend at least reviewing these. And there are some really basic QC checks. You can just look at the signal, the features of your data, and get a good idea about not only do I have a bias, but maybe what that bias is. And talking to your genome center, your colleagues, you might be able to actually work out what you can change. Okay, so FASTQ quality is always essential. We have this in the ChIP-seq course, so I don't think we're going to cover it here. But just a reminder, we do also have this wonderful package. Hopefully I'm still sharing screen. So, you know, Way has this lovely package, rfasp, with a capital. And if I change my working directory, set working directory, choose directory to my R course. This is a really simple program just to give you some very quick quality control. Data. Then I just have to give it an output file. Produce these nice little JSONs for us, right? Where we summarize all our quality across our things. You know, this data is probably pretty good. It got published. We've shown you methods of how to do this in our interrogating the read separately, but I think this rfasp, as you saw, it's very fast and it gives you a nice HTML to review pretty quickly. So I'm a little biased, but I always recommend using that. Assuming all your data looks good, we can continue. In this case, it did. And here we're going to process one sample from the green leaf data. And it's a little unfair because this really is the first data set, but it does perfectly capture some of the features of ATACseq. So we're going to use this and we're going to process this. So first thing we're going to need to do is create a reference genome. We can do that from our bioconductor objects. So we know, I'm not sure if this link still works, hopefully. Great. So we know we have BS genome packages and BS genome packages in Bioconductor. They are the container, the annotation package, which holds all the reference data. Okay, so we know we can extract full sequences for every chromosome from these BS genome objects. In this case, I'm going to work at HG19. I think that might be, you know, HG38 is the latest genome. HG19 is the one before. Maybe that's to match what they'd aligned it as. So here we're just going to take the library, we'll load it. We're going to take the main chromosomes, for example, here. So we'll take chromosomes 1 to 21, X, Y, and M. And then we're going to loop through these main chromosomes, and we're going to extract, because we're looping through the names. For every time we loop, I'll apply. We have a name here, because we're going through the chromosomes, and it extracts the sequence for every chromosome one at a time, and it puts it in a list. Then we have the list. We want to assign names to it. So we have our named list of sequences. And then finally, with a named list of sequences, and we want the names because these are what the contigs will be called. And when we align, these are what the chromosome names will be. So with that, we're then going to create a DNA string set, right? DNA strings would just be one chromosome. This is a DNA string set. And then we'll write this out to file. I think that was just make sure this all works. It'll take a second, I think. It takes a second to extract all this, but a lot quicker than having to download the full genome. But we did show you if you wanted to, with all this, you could still go somewhere like iGenomes and download all the chromatin. Maybe that's going to take a few seconds longer. Okay. What is important, though, is that at the end of this, we have a DNA string set. We write this out with write string set. This gives us a FASTA file because we've said .fa at the end, and FASTA files, as we saw. This is how we contain our reference genome information in a text file. So the next thing we're going to use is our subread. Did our thing build? Okay, great. So we add a DNA string for every single chromosome. And then I think we made this into a DNA string set here. And once we had that, we can just write that out with this write X string set and start doing our align. Okay, so we use, I think we're going to have two versions here. I think this is because you know, our subread used to be just Linux and Mac, but I think in the last releases it became Windows as well. So this is a really fantastic aligner. As we said previously, we're not going to do, we do the quality control here, right? Our QC, we can do this in FastP, all the methods we showed you in our bioconductor courses. But we're not actually going to remove anything from our reads. We're going to maintain them. We'll just take the quality. And that's because, as we showed you when we did our bioconductor intro, our subread has fantastic ways for dealing with, you know, adapter contamination, any problems with your reads. It's a very robust aligner. Before we do anything with our subread, we need to build our index. So we need to create a file, which is a little bit more useful than this FASTA file, for our subread to start to do its alignment. We only ever have to do this once per genome. Because once we've built it, we can just carry on using the same index. So in this case, we're going to go build index, or load our subread, build index. We'll give it our BS genome object. Sorry, BS genome FASTA file here. So the actual FASTA file. We'll tell it the name of the index we actually want to create. And then here, to save a little bit of space, a little bit of memory, we're going to set the memory to be 1000 megabytes. So that's quite small. It's not going to use much memory in alignment. But we're also going to set an additional parameter called index split. And this actually creates little gaps in the index, which it claims, and I think it's shown, that doesn't affect the alignment. So we can actually save a little bit of more memory here as well. So now we have this index, right? We create our index. We can start to align our ATACseq reads. So I think previously, or up to this point, we've only been dealing with single end reads, right? ATACseq is typically paired end. It's not always paired end, but typically it is. So we need to make some minor adjustments to our alignment steps. Typically, when we get our aligned data for paired end reads, you're going to have something like _1, _2, or _R1, _R2 to signify that these reads are pairs, right? So here, back from the Genome Center, or in this case, the one we downloaded, you'll get your SRR 891269_1. This is our first read in a pair. And then this is our second read in a pair. But these files are totally related. They shouldn't be used independently. They're just split into two. And now with that, we can actually start to look at some of these properties. So our two matched paired end read files will most often, hopefully always, contain the same number of reads, and the reads will be ordered the same in both files. Okay, they really should contain the same number of reads. If they don't, something's happened to the files. They've either deliberately been edited or something's been lost. They should be in the same order as well. So we can check things like, will the read names match across our paired files? And this is really going to signify that they're in the correct order. It also just demonstrates a little bit about how these work. So we can use a short read package here, require short read. Short read is our package to deal with short reads, FASTQ, reads in FASTQ format. We use the read FASTQ. We can read in this gzipped. We read in the second read in the pair. So read 1, R1, R2. If I look at the ID of the first two reads, you can see they should, let's just double check, they should match across the two, right? The only difference being there's 1, and here we have a 2, right? But the reads have come from the same location, the same flow part of the flow cell, right? It was just, this was the first read part of the fragment to be sequenced. And then the second part of the fragment, the other end of the fragment was sequenced. It was given the same name, so we know where it came from and we can tie them together. It just told us 2 or 1 to allow us to know which side of the fragment we're on. Okay, so this is really what a paired read looks like, I guess. So we have our fragment, which is actually this middle portion here, right? So this is actually what we're sequencing. And then we have our adapters, which we use to stick to the flow cells, and we start to do the sequencing from. But what we're going to sequence is this read 1, and then in a second stage, we'll sequence going the other way, read 2. So then we have some terminology just to look at here. So we obviously have the adapters on the end. We have the insert in the middle. So this is the bit we actually want to sequence. And that gives us something called the insert size, which is basically the size of the usable fragment. So this is often interchangeable. People will refer to this as fragment length, me included often. In reality, the fragment length, what you actually do see when you do a gel is the insert plus the adapter. Okay, that's the actual fragment length. And finally, we have something inner distance. Okay, and this is the distance between the end of the reads. You can imagine that the inner distance can sometimes just not exist, right? If you have a long enough read and a short enough insert size, read 1 will often overlap with read 2. Okay, sometimes you can have a read which will go all the way over read 2 and into the adapter, right? And this is where we have things like worries about adapter removal, because we've sequenced all the way through our insert and into the adapter. Okay, again, our subread doesn't need to worry about it. It has methods to clip out adapters as part of the alignment. Okay, but this insert size here, this length of this fragment is very important to us, because actually, I'm not sure if I show this, because this in ATACseq allows us to know whether we have cut around smaller things like TFs. We would expect a smaller fragment, right? If we're cutting around the transcription factor site, it's a smaller thing. A nucleosome has a very defined size. So if we can use things like fragment length to say, okay, once we're above a certain size, we're most likely coming from nucleosome-like fragments, cuts around nucleosomes. So it can be very useful to us, this insert size distance. It tells us, what type of fragment, where from these open regions are we coming from. Because this is not RNA-seq, this is a contiguous alignment to the genome, we can use standard alignment for DNA to align our ATACseq signal to the genome. One thing we are going to change is to increase the maximum allowed fragment length. And this is just going to allow us to capture fragments which haven't come from just one nucleosome, but when 2 nucleosomes are quite tightly together, maybe we are now around 2 or even 3 or even 4. So we're going to take a longer maximum fragment length just to allow for the fragment length we know they had in the paper, in their Greenleaf paper. They claim to get fragments up to this length. Okay, so we can control that by just setting the max fragment length here. We're just going to set that to be 2000. We haven't set min fragment length. We're also going to set a new parameter, unique. So we're only going to get reads which uniquely map. Remember, there are places in the genome which just aren't unique. 100 base pair sequence on chromosome 2 may appear exactly on chromosome 7 the same. If we align something here now with this unique parameter, we're just going to throw it away. Okay, we can't trust where the alignment really went anyway, so we're going to ignore them at this point. Often it's not a big problem to just keep them, but for ATACseq, at some point we're going to want to remove multi-mapping reads. To use bow tie, we do a very similar process. So I said our subread, this will give us our aligned BAM, and then we know we had to do just a couple more steps to get to a BAM, which is useful. Sorting, indexing. Originally, our subread wasn't available on Windows and Linux, so we did consider how we could use our bow tie 2 here. And it works well. Our bow tie 2, again, is a DNA aligner. It aligns to contiguous genome. It's not going to take into account splice sites. So we can just load the library rbowtie2. We give it the same reference file we used for our subread, our FASTA format, written out DNA string set from BS genome. FASTA format is absolutely the standard for reference files. If you want to use star, if you want to use HiSat, GSNAP, any of your aligners, you're going to need to give it a FASTA file anyway to make an index. And then we just give it what we want to call the index. So it's going to create I think in this case, a folder full of files it needs. Leave that and it'll go off and create our index. Again, we just need to do this once, but we need to do it once per program, right? RBowtie2 works differently to our subread. Therefore, it wants to catalog and index the genome differently between the programs. The pain with RBowtie2 and Bowtie2 generally, it doesn't accept fastq.guz, so it doesn't accept zip files. Actually, when you're working on the command line, this isn't so much of a problem. You can just pipe them in R. It doesn't work like that. So we need to unzip them first. Maybe someone knows better than me, but I think it's impossible to do gunzip while we're piping it in, or at least it's not very pretty. So here we're going to do gunzip on our 2 files, and this is just going to unzip them, but unzip them for this gzip, gzip version of zipping up files. Once we've unzipped that, now we have this fastq file, no longer a GUZ. We can start to align it. We just need to specify to our Bowtie2 function, our index, our output file, and the 2 sequence files we've just gunzipped, unzipped. Slight pain again with Bowtie2 is it needs to make SAM output. It's not going to make us a BAM output in one go. So we need to be able to do that ourselves. Luckily, our SAM tools, which is our wrapper for SAM tools in R, we don't need to read the file in to do anything. We can just apply as BAM to that SAM file. And it's going to create a SAM file with a BAM file with the same name, but with .bam at the end. Okay. It seems daft, this unzipping and then turning it back into a BAM. Remember, Bowtie2 was written for the command line originally, and it's been wrapped in R. All these things like creating the SAM file, usually you would have an operation in Linux on your command line, which would just pipe it straight into making a BAM file. So it looks painful here, but actually it's way less painful on the command line. But still, only a few extra steps, and you can align everything in RBowtie from inside R2. I still prefer R subread. So following alignment, we would want to sort and index our BAM file like we have for every other BAM file we've produced. In this case, we're going to sort it by coordinate. There is sometimes with paired end data, especially paired end data, reason to sort it by read name. And that's because the read name, as you see here, between read 1 and read 2 only differs by this bit here. So once you start sorting by read name, actually you can arrange your pairs together, which can be very useful. You want to know pair 1 was here, pair 2 was here. Some programs really want that. In this case, we don't need to for working in R, so we're just going to do a standard sort. We're going to create a file name here from our outBAM, sorted.bam, and then we'll just write out to that file, sort our BAM file to this file here, but I'm removing the extension .bam, right? Because we know that this is always going to write out add.bam to the end of the file, and then we can index it. So just to repeat that, every time we produce a BAM file, our subread, RBowtie, we can check the sort order, and we saw that previously, but generally we're going to want to sort it here by default coordinate sort, and then we're going to index it, and it's at that point we can start to do our random access, things like IDX stats now, but also view it in IGV. Use other programs to start to get counts from our SAM file. So now we have a valid BAM file we can start to operate in other programs, only after this bit. First thing we can do, because we've indexed it, is use the rsamtools function, which is based on the samtools function, IDX stats. And we just use this function, IDX stats BAM, from the rsamtools library. We give it our sorted BAM file, and it's going to give us back a data frame. Okay. So with that data frame, we can start to do our plots of here. I'm going to load ggplot. I'm going to take our data frame, and it's going to be chromosome, number of reads mapping to it. So seq name, number of reads mapping to it, and then the length of it. If here we plot our seq names across the Y, our mapped number of reads mapping to a chromosome along the X, and just do a bar plot, we can already see potentially a big problem. Right? And this is just an artifact or a issue with ATACseq, which existed at the beginning but still exists now. You get a ton of mitochondrial reads. Okay, and we're talking, if this is based on size, chromosome 1, you know, it's 10 times, 20 times, 30 times the number of reads landing in chromosome 1 went to chromosome M. Actually, you can filter out chromosome M and have great data. I think the real issue here, and the real reason people are using things like OmniATAC, which washes away this mitochondrial reads, gets rid of the mitochondrial contamination, is because if you're going to sequence, you don't want to spend all your money on chromosome M. Unless you've got some particular interest in mitochondrial chromosome, and maybe you do want to spend all your money on that. But most people don't. I'm not sure of the quality of the data across those mitochondrial chromosome anyway. If you're going to throw that all away, you'd rather do that before you sequence it all, because sequencing is expensive, and this is often the first, when people come work with us, this is the first thing that they want to check. How did I do on this? You will do better than they did on the first paper, clearly. I mean, it's been optimized, and I think we're going to look at Christina Leslie's in the exercise, and it just looks so much better. Not gone, but so much better. Okay, so now we've processed the Greenleaf data set. Right, so this is all the original FASTQ data set. And we can start to actually work with the alignments. What can we actually do with these alignments? So first thing we're going to do is identify the expected, and I say fragment length, and as I say, I use this interchangeably. I mean insert size here, distribution. Okay. How long is my actual sequenced fragment? Not the adapters, but the internal bit. So we can actually read our newly aligned data, these BAM files, using this genomic alignments package. So we should have seen that in the last day of my bioconductor sessions last week. If you didn't, we're going to show you now anyway. We don't want to read all the data in. Potentially that's going to cost us a lot of memory. We're on our laptops. We don't want to break them, but also we don't need to, because we only really want to consider at this point properly paired reads. Okay, so properly pair is actually a flag in a BAM file, so it's a binary yes, no, we are properly paired. And we're going to do it by setting our scan BAM flag inside our scan BAM param to be is proper pair true. Okay, and this means that when we set our max fragment length to be 2000 BP, anything underneath that will be considered a proper pair. Everything longer or you know, one read aligns to one chromosome, the other read aligns to a different chromosome, we're going to consider that an improper pair, right? And we're going to throw them away. To do that, we're going to use the scan BAM flag. We're just going to set is proper pair, and that's going to give us, I think, a scan BAM flag object. And then with that, we can create a scan BAM param object. So we're going to pass in our flags. So this is the scan BAM flag we have here. We're going to pass that in. So we're only going to get properly paired. And now I want to say what I want to get out. So if we go back to our genomic alignments course, we know when we read in genomic alignments, it's not going to read in all the information from the BAM. It's actually just going to read in start, chromosome start, end, and some information on the cigar string, right? How it's aligned. Did it align continuously? Or did it align in gapped, splice across things? It's genomic alignments, right, package. So it's just going to really take the information it needs for the alignment. In this case, we want more information. And actually, if you want to do things like max 2, repeat calling, you need a little bit more information in your bound file just because it is checking for it. So we need to include a bit more. So what we are going to get here is QName. I'll go back to R to show you a little bit about this again. QName, mapQ, so the name of the read, the query name, the mapQ, which is the quality of the mapping, and importantly, isize, which is the insert size. So we want them back because we want to start to make statistics from them. They're in the bound file, so read them. We're also just going to read here, just to save us some time, chromosome 20. So I've looked up the length of chromosome 20, so I know it's 1 to this position. We can get this from the seq length too. So to do that, I just need to create a GRanges showing the limits of chromosome 20. And I provide that as my which. So now I have for scanban param, I have a what. So it's going to bring in these additional features. Read name, mapq, insert size. It's going to go which. It's only going to look over this region in the genome. Remember, we can do that because it's indexed. So it can just go to that region straight away. And we're also additionally going to set the flag. We just want proper pair. So this is now our scanban param object. You can see here, there's a class. We have our one flag. We have our one range. And we have our QName, mapQ, and isize as additional what to bring in. Okay, so now we have our scanban param object. And just maybe I'll just hop out for a sec. I think scanban param is super powerful. If you really require our SAM tools, I think I showed this in the Bioconductor talk, but just to show it here as well. So scanban param, look up the help for it. It tells you some information on what you can choose, how you arrange this, but actually it didn't really tell you what you can, much about what you can get. So like, how would you know what QName is, and how would I know isize is the one to select? If you go to the help, you can trust the help here. It does go to the what, and it says the fields are described in scanban, which we're not going to cover, do we? I don't think we cover scanban. It's very low level interaction with BAM files. But here, if we go to the, somewhere here, yeah, we have value, and you have all the descriptions of the QNames, the flag, the R name, all these things are what you can bring back in. The sequence, we don't have the sequence, do we? All these things we can bring back in from the BAM file, but by default, we won't, and it will save us time and memory. Okay. So we have our reader giant. We've seen the read GA alignments before for other types of files, BAM files, when they're single end. Now we have to do something a little bit more complex. We have to use read GA alignment pairs. So it needs to be aware of the pairs in the BAM file, but it takes the same arguments as read GA alignments. We give it our sorted BAM file, and we give it our newly created scanban param object, and then it goes in and just reads that bit of information we need. Okay. So that shouldn't take too long, and we'll get back a GA alignment pairs object, but still from the genomic alignments package. Okay. So in contrast to what we've seen before for a GA alignment object, a GA alignment pairs object is actually a very sensibly, basically 2 GA alignment objects attached together, right? One for each read. Okay, when we look at it inside the actual RStudio browser, we can see attack reads 1 to 2. So I'm just looking at the first 2 reads here, what we just read in. We can see it also displays quite differently. Okay, just save space inside our screen. This is just a display. But if we write this into the console, we're going to get chromosome 20, right? The seq names are on the same chromosome, the strand, and then we have the ranges for the first read, and then the ranges for the second read. Okay, so this is my first read in the pair, and then this is my second read in the pair. And they're exactly the same position, actually, these first 2 reads. But okay. As I said, these are actually 2 GA alignment objects put together. And if we want to get access to each of these objects separately, We can either use first or second, and they do just like they say on the tin. They're going to give me the first or the second read in the pair. Okay, like we had R1, I get that with first. R2, I get that with second. If I do first on my GA alignment pairs object, I get my RG range, my GA alignments object. If I do second, I get my second GA alignments object. And now we can see something we're more familiar with, where we have our GA alignments object, chromosome, strand, cigar. So this is 50 matches in a row. The query width, so the mapping of the read to the genome, how long that actual map is. No, sorry, that's the mapping of the chromosome to the genome. This is actually the length of the read, the start map, the end map, the number of junctions. Here's 0. Right, no, so we know it matches 50 in a row. So we don't have any junctions. Importantly, we now have the Q name. So this is the read name. We have the map Q, how good quality the actual alignment was. And we have the insert size. Okay, so how big is this portion of fragment we sequenced? Okay, so first thing we might want to do is just get the distribution of MapQ scores. How well are these mapping? We can extract any information from a GA alignments or a G-ranges object from its metadata columns. Here it's got 3, using the mcols function. So we'll use mcols, give it the read 1, and we'll extract, just like with a dollar data frame, $MapQ. So that's going to give us this column here from read 1. And then we can get the read 2 column as well. And I can see I've got 2 40 40. To get this, or to try and summarize this for any type of plotting, I can use this function table. Okay, so table will tell me how many times a value occurs in that list vector. Sorry. And here I can see I have 264,000 40s, so high quality alignment. And I have 252 very low quality alignment. And it's a similar distribution for read 2. So if I can then take our ggplot magic, string this together from our MapQ named vectors here, I'm going to take the names of those vectors, stitch them together with my read 1, read 2, create at the same time like another column which will tell me whether these were read 1 or read 2, whether these qualities came from there, and then I attach the frequency as well just off the screen. So effectively here I get a data frame, I get a MapQ column, the actual quality, I get the number of times I get that quality, and I get whether it came from read 1 or read 2. Gonna then change the factor levels just so that they're in a nice order rather than in a random order. And then I can just make our ggplot. X being our MapQ quality, getting bigger, Y being our frequency, fill them in by MapQ, and then just do a bar plot. Yeah. This looks surprisingly good, to be honest. Both our mappings are high. You'd be more concerned if you saw a bump here around 8. So if you saw some reads which are very low quality mapping, this suggests that either you had a very low quality data, the FASTQ going in, or you're actually mapping to regions which just can't be mapped well. So highly repeated regions, these are regions we can't map well. And they will often, so highly repetitive regions will often be a MapQ of 0 in most cases. Okay, so we've seen how we can look at MapQ. Perhaps a more or most useful metric we can get from GA alignments is to assess this fragment length, these insert sizes. And that's because we will see an effect in ATACseq data on the insert sizes, which really tells us whether or not we have good ATACseq data. So we want to be able to plot the distribution of insert sizes. So we saw that insert size was something we brought in, and we deliberately brought it back with our what. So we just need to be able to extract that we can extract that from either read, right? Because the insert size will be a mirror for read 1 and read 2. They're going to both have the same insert size because they were part of the same insert. So in this case, we're just going to take the read 1, and we'll do first, and we'll pull out the reads for read 1. And then we'll extract from there the I size from our read 1. So we're going to, I use element metadata here, but you could use mcols. They do the same thing. But I'm just trying to get this column. And then with that, I'm going to make them absolute. Right? So I don't care if read 2 was this direction or that direction. I only care about what was the absolute size of that insert. And then we can just look at the head here, and I can see I have some distribution of sizes. I have a very small one. I have some quite long ones. I have some medium ones. Again, we can use this trick of table. So if we give our insert size, vector of insert sizes, and this is going to be a big vector because it's all our reads. If I pass that to table, it's going to then produce a nice table of, I got this many at 50, this many at 51, this many at 52, etc., etc., etc. With that, then I'm going to create a data frame. So I can do some ggplot. So we're just looking at chromosome 20 here. Could have done this across multiple chromosomes. So first thing I do is take my fragment length sizes, and I want to take these names. So anytime we have a name in a vector, it's always a character, right? So this isn't numeric anymore. It looks numeric, but it's a name, so it's a character. So we're going to go names from them, and then we're going to turn them into a numeric. Right? We don't want them to be a character. And then going to get the count, so how much we have of anything. And again, that will just be the fragment length sizes, probably not necessarily, but making sure they're numeric there. And then we're just going to get a data frame of the different fragment length sizes and how many times a read was that size. I take that then to ggplot, and we do our insert size along the X, our count, the frequency along the Y, and we'll do a geom line. And it doesn't look super great yet, but we do see, if you really squint and look at the bottom, once we do this fragment part, give it a theme BW, you can see suddenly we have lots of very small fragments, but then you can see this bump here at the bottom, then maybe a second bump, and there is a third bump if you zoom in. Okay, so actually these bumps here represent our buildup of fragments around nucleosome size fragment sizes, right? If we put this onto a log2 scale, actually now we can start to see very clearly bumps here which correspond to the size of a nucleosome, 2 nucleosomes, 3 nucleosomes, 4 nucleosomes, 5 nucleosomes. Okay, but we need to put this on a log scale. So to do that, we just do fragment length. We can now just apply scale y continuous, trans, let me check the chat for a while. Trans y continuous, trans is log2, theme BW. And now we can really see this presence. Okay, and this is because the ATACseq has got around a nucleosome. Right, and those times it's got around 1 nucleosome, we see lots of fragments roughly this length. When it gets around 2 nucleosomes, we'll see fragments this length, 3, 4. And clearly this happens more often than this. Anything less than this is going to be all transcription factor, signal around transcription factors rather than signal around nucleosomes. So actually in the Greenleaf paper, I mean, this image is identical to what they produce. So in the Greenleaf paper, they annotate these, and they say anything under the green line, 1,000 base pairs is our nucleosome free, right? No nucleosome, maybe transcription factor binding there. Our 180 to 247 would be our mononucleosome. I think a nucleosome is 147, but I guess they set above this, right, to make sure you captured it. Dinucleosome 315 to 437. And we can just add to our plot some V lines to just show this is what they considered mononuc, this is dinuc, and this is our nucleosome free. What's really nice is, you know, with our method here, using our subread, which is a different method to what they used in their paper, we can recapture their results almost exactly. right? Just by the methods we've learned in our last few weeks in Bioconductor, we can at least get the same QC, and we can at least get BAM files which look pretty darn similar. Okay, so some of the next things they do here in their paper, I actually don't do this too much in our own work, which is splitting up our ATACseq signal now into mononuc, dinuc, and this open regions, nuke-free, because they're all open regions. So we're going to show you how to do this, and it is useful, and it actually can be kind of fun to look at your data and separate it out. It seems like most people just include all the data, assuming that it's all open, right? All coming from open regions. But we can do this here anyway very easily because we have our attack reads, right? So this is our these reads we read in, right? So these are still the pairs. And we can actually just use the insert sizes because it's still in the same order, right? We can use the insert sizes vector and just subset. I want those less than 100. And we've made this absolute at this point. Okay. So we can go, you know, they've got to be either less than 100 or greater than 180 and less than 240, or greater than 318 and less than 437, right? Based on the Greenleaf kind of definition of insert sizes, we can then subset our attack reads into these groups. And then if we wanted to, I'm not sure we've actually done this yet, we can actually export the BAM file. Maybe we have. We can actually export the BAM file using our track layer. We just need to give it our subsetted genomic alignment pairs. And now we can export our nuke-free region BAM. Just need to specify format as BAM. Export our mononuc and export our dinuc, again, format as BAM. Okay. So you can actually read in your BAM files, split them out, separate them. You can even manipulate the reads in them, and you can still write them out as BAM files and use them in further analysis. Okay. We can, this is kind of nice, we can create full length fragments from our single end reads to evaluate duplication rates. Okay, how duplicated is not a read position, but the full fragment, right? Because a read 1 might be in the same position across a few fragments, but the read 2 would allow us to differentiate these came from different fragments. So we can actually make our attack reads into a pseudo kind of single read running all the way from read 1 to the end of read 2 by just applying the granges function to our GA alignment pairs object. Okay, so here we have a read 1 which goes from 6,000 to 60,410. Right, so here I'm going to do granges on that GA alignment pairs object, and now I don't have 2 reads, I just have 1 read, and it goes from there to there. I don't really have a read, to be fair. I now have a granges, but I now have the representation of the whole fragment rather than just the ends of the fragments. Okay, so then we can use our duplicated function to get an idea then of what granges are duplicated. Where do we see in exactly the same position in our genome? So I can get total duplicated fragments by doing some duplicated attack fragments. And then I can get the length of my total, whatever granges I have, my total number of granges. I can get a duplication rate then by doing my duplicate fragments over my total. And then my non-redundant fraction is 1 minus that. And the reason I say non-redundant fraction, this is a very standard QC measure for ATACseq. It's kind of nice that we can do this in Bioconductor just in a few lines. We can make our granges into a single fragment. We can then use the duplicated granges function to tell us how many times do we see in exactly the same range, get the sum, and then get our non-redundant fraction, which here is actually pretty good. This means that 26% of our reads were identical positions to another read. Okay. We can make it a lot quicker to actually review our pileup of a ATACseq signal in the genome by creating this bigwig file. So this is the binary version of a wiggle file. So to do this, we can take our G ranges of full fragment lengths we have. We can try and account for the fact that we may have different numbers of fragments across different experiments. So we're going to normalize it. So what we'll do is I'll take our TAC fragments, we'll pass them to the coverage function. Okay, and coverage is going to turn this into an hourly list. So, number of fragments at every base pair. But we're going to additionally apply a weight, which is just 1,000,000 divided by the number of fragments we have. Okay. We then use export bigwig, give it our newly created hourly list, give it our open region bigwig file name. Here I'm just going to rename our BAM to this. And then we'll get a file here, which we can then read in IGV. We can start to manipulate. We can use with our other bigwig tools, to get some coverage, etc. But in a few lines again, we can go to something where we have a normalized, sensible bigwig we can start to review. So I think I just have a few more lines here. I just want to mention a package, which is good and has lots of metrics available in it. I think we have covered what I would say are the most useful up until now. There are a few more useful coming up once we've done some peak calling. But I'll leave it up to you to go and review this package, ATACseq QC, and see if there's anything I'm missing here. We do use this in our pipelines. There's probably one metric I don't include, but I think we have an approximate to that in something else I'll show later. But two really useful functions which can come out of this are PBC1, PCR bottleneck coefficient 1, and PBC2. Okay, and these are used by ENCODE. So we have some nice guidelines that you can go and have a look in their ATACseq guidelines, but you need to be able to generate them. So we're going to use ATACQC to do this. This is a bioconductor library again, so we just Google ATACseq, whatever, it will take us to the page, and we can go and copy and paste, or we do BioC Manager install ATACseq QC, and it will go and download all the dependencies and give us the file. So as with ChipQC, our package ATACseq QC will do a lot of the work in one big go for you using a workflow function. And it's going to go off and do all these operations and then just pass this back to you. Okay. This, I say this is an understatement, this can be fairly memory heavy. Some of the BAM files which we have to work on, we run BAM QC in our data set, in our pipelines. It really does run out of memory, and it will die even on high performance computing because it's just not written to be memory efficient. What we have found, and I'm going to just do chromosome 17 here, I have found that downsampling our BAM files to around 40 million reads makes it still possible to do this BAM QC, because some people come with billion read files, but still get the sensible quality control statistics. So downsampling to something like 40 million allows you to maintain the characteristics of whether it's good quality or bad quality without having to read a billion reads into file. So in this case, we're just going to, I've just already pre-filtered this to chromosome 17. I may update this in the future to show you, but we did cover this, how you can just subsample your BAM down to a certain number of reads. Right? Here we do ATACseq QC, BAM QC, very simple function, just takes your BAM file. It will go off and spend a few hours doing its QC. If you do this on the cluster or on your laptop overnight, you're not going to worry. And it gives you back an ATACQC object, which contains slots of QC information, and a lot of them we've already covered, I think, in reasonably easy ways. So duplication rate, we have proper pair rate. We already showed you how you can just read in proper pairs and get this. Mitochondrial rate, we saw this in our plot earlier on. You know, unmapped rate. We got this when we ran our alignment. MapQ, we did ourselves. IDX stats, we did ourselves. Non-redundant fraction, we just did ourselves. But PCR bottleneck coefficient 1 and PCR bottleneck coefficient 2, we could calculate themselves, but I think this is good to use this package, and it's a nice example to show you its use. So these really identify this PCR bias over amplification, which can occur not just in preparation of ATACseq samples, but also ChIP-seq samples, anytime there's really a PCR step. So the first one's PCR bottleneck coefficient 1, and this is calculated as the number of positions in a genome with exactly one read mapped uniquely compared to the number of positions with at least one read. Right, so exactly one read, where we just have one read, versus the number of positions with at least one read. So an example, we have 20 reads, 16 map uniquely to the locations, 4 do not map uniquely. Instead, there are 2 locations, both of which have 2 reads. This would lead to a calculation of 16 over 18, and we get this PCR bottleneck coefficient. What's really nice about PCR bottleneck coefficient, probably why I really recommend its use, is that there are standards in the ENCODE we can kind of look at and compare our quality to. Okay, so values less than 0.7, according to ENCODE, would indicate severe bottlenecking. Between 0.7 to 0.9, moderate bottlenecking, greater than 0.9, so no bottlenecking. And when they say bottlenecking, I'm not sure I've described this here. This is just this idea that you have, oh yeah, it's called bottleneck coefficient. It's just this idea that you have, you start sequencing the same sequence over and over again. You're not actually really generating more information. You have a few sequences which win, and they take over your amplification. And when you go and sequence this, you have a very low complexity library. A few positions where many reads map and not many where only one read maps. So PCR bottleneck 2 is our secondary measure of bottlenecking, and it's calculated as the number of positions in the genome where exactly one read mapped compared to the number of positions where exactly 2 reads map. So previously it's 1 read versus more than 1 read, so 2 or above. Here it's exactly 2. Right. Similar metric, probably not quite as useful as 1, but again, we have some standards to compare to. Values less than 1 indicate severe bottlenecking. The 1 to 3 indicate moderate bottlenecking, and 3, no bottlenecking. Okay, so here we can see in our sample, we have 3.73. So we're not too worried about the quality of this data. This was indicate moderate, but this seemed to indicate none. So we'd be happy and we would proceed. If you came in under these values, you would start to maybe be concerned. That said, you know, if you came in, because this is based on number of positions with 1 versus, you know, multiple, this is affected by how many reads you actually sequence. So if you sequence 1 billion reads, you'll get slightly, you know, crappier PCR bottleneck scores. Okay, so we got to the end of how you align, how we can go from FastQ data, paired FastQ data, through aligned alignment to our bound files, and then how we can start to get some of the most important QC of your data. Principal things to watch out for when you do your ATACseq analysis is to get this graph. Okay, I think once you see some bumps in your ATACseq data, you can be pretty comfortable that you've got nice quality ATACseq data. Another major point to review is this chromosome M. You shouldn't be anywhere like this. The examples I'm going to show you now, the methods, the exercises, you'll see this is much lower. The final QC metric is going to be the same as with ChIP-seq, which I find really useful, is read and peaks. But we're going to cover that in the next section once we've done some actual peak calling. So we have some QC here. I think this is actually from, this is now where we use Christina Leslie's data. So, you know, it'd be interesting to go and have a go at this. I think I do provide BAM files. So if you want to, you can skip ahead. You don't need to do the alignment. You can actually just go take the BAM and start to see some of these quality we just reviewed now. How does this look in the BAM file? And, you know, and I can tell you, quality got a lot better in those few years. So we're on our page. We're on our ATACseq with Bioconductor within our Rockefeller page. In our last session, we learned a little bit about what ATACseq is, but we learned how we can go from FastQ. If we wanted to, we can do QC, either using our standard methods or using rFastP, which we can do that in one line, gives us a nice HTML report, and we'll add to the courses soon. We saw that actually with that, we learned a little bit about paired end data, both how we can align it. We just need to specify in the align parameters, arguments that we have a second read. And then we saw it's a little different when we get to the alignments. We want to read this in using genomic alignments. We're going to get this GA alignments pairs. And that is just two really alignment objects side by side. So we showed how we can deal with that. And actually, most importantly, how we can extract QC, useful information about how good our ATACseq is, and how we can create our fragments and visualize them in IGB. So in our next session, we're going to go about actually calling some regions we would say are open. So we're going to apply a filter using max2 to say this region has more signal from our ATACseq data. So it's probably more open than we'd expect by chance, more signal than we expect by chance. It's likely to be an open region. And then we're going to do some plots. So actually looking at our mononucleosome and nucleosome-free proportions of our signal, some plotting of cut sites around DNA, binding proteins, but most importantly, we're going to do some annotation. We're going to go, this is an open region, and this is the gene it's most likely regulating. Okay, most likely, because it's really hard. You can't be 100% confident in anything without something like high C. Probably not 100% confident now, but we know this open region may regulate a gene. So in this section, we're going to work more with the greenleaf data. You can go and remind yourself how we processed this before. We're going to look at things like TSS signal, ATACseq. We expect TSSs to be quite open. So we should see a lot of signal there, and we can actually use this as a measure of quality. We're going to use max2 to actually identify regions we believe are significantly open and annotate those to genes. And then we're going to end with just doing this transposase cut sites. In our last session, I'm not sure I'm going to show it here. Here I am. We show that like this is where the actual transposase cuts. You know, if we get enough resolution, we should be able to start to see cut sites around things which are protected, such as transcription factor binding sites. And this will allow us then to see things like changes in the cutting around transcription factors. Maybe we could say, oh, that transcription factor is not so active, it's not binding so much in this sample. Okay, so we have data from ENCODE as well, liver, kidney, brain. This will allow us to do differentials, and I have processed some of this. So you can actually download the BAM file and the BAI file. I think we got a link here. This is a big download. I just downloaded it in the graph. It's around 2 gigs, but it contains all the files we're going to talk about. So, you know, if you want to skip ahead, join at certain parts of the processing, it's all in there. But really, sorry, this is essential. Everything, the full data set, everything which can be processed in this data set, you'll find here and here. things you need really to work along the course are actually just in here. So to remind us, ATACseq allows us to get signal around regions which are protected but are in open chromatin. Okay, so here we have a little bit of an open nucleosome and we can get some signal around it, but around transcription factor binding sites, we can pull out sequences once we've cut with this transposase, and then we align them to the genome, and we have an idea about which regions of the genome are open. This does mean by having this cut, and we can actually extract signal from around transcription factor sites and around nucleosomes, it means, as we saw in our kind of fragment insert size length plot, then we have mixtures of signal types in our data. We have signal coming from nucleosomes, and we have signal coming from nucleosome-free, most likely transcriptional machinery, transcription factors binding in that TSS region. And actually, I just split out some data here. I think this is the data. Oh, maybe I've taken this from a paper, to be honest. I'm not sure. But here we have ATACseq nucleosome-free region. So if I take all the reads which are less than 100 base pairs and I make a bigwig out of them, if I take all the reads which were nucleosome length, the fragments which were nucleosome length, and I plot them, I start to see signal where I might expect to see nucleosomes. We have the +1 nucleosome here, or is it -1 nucleosome? The nucleosome sitting between our TSR genes. And here we have like the transcriptional machinery assembling at the beginning of this transcription, this gene. Okay, so already we can split our data into these two and start to investigate that visually in IGV. The shorter fragments should represent, as I say, these open regions, right? They should come from these regions right at the TSS. As we actually see here for one TSS. Our longer fragments, these slightly 180 to 240 something, these are going to represent signal fragments coming from around nucleosomes. So we should start to see that signal not directly on the TSS, but maybe at the +1 nucleosome, which is often very strong, and the negative nucleosome here, the -1 nucleosome. So, this is exactly what we're seeing here, +1 nucleosome, +1 nucleosome, -1 nucleosome. Often this is a very strong signal. So we can use this expectation. Again, there's some type of quality control for our data. If we have good quality ATACseq data, we should be able to separate it out into our non-nucleosome signal, our nucleosome signal, and expect to see a pattern of signal around the TSS similar to pole 2 for the open non-nucleosome signal. And then for the nucleosome-like signal, we expect to see something like this, where we see a +1 nucleosome, a -1 nucleosome, +2 nucleosome, +3 nucleosome, and it should trail off. This is really perfect data I'm plotting here, so we may not see something quite as nice as this, but we want to see our nucleosome-free proportion at the TSS and our nucleosome signal proportion at the +1. Okay. So what we're going to use is this idea of a meta plot. We focus on the TSS of a gene, and we kind of pile up all the TSSs on top of each other, and we create an average signal across a TSS for our bigwig signal, our coverage from our experiment here, ATACseq. So we can use another one of our packages, Soggy, terrible name, in order to make these plots. Soggy is a bioconductor package, so you can install it either by Googling it, and it'll tell you, where to go, copy paste, it will work. Or we can just do it here, BioC Manager, install, and give it Soggy. It's meant to be something like summaries over group genomic intervals, but it uses ggplot, so we put it in the name. we can load this then, and this gives us access to all the tool sets available to allow us to do these types of plots. Okay, so the Soggy package, it really is a terrible name, the Soggy library requires a BAM file. Okay, it also accepts bigwig files, which now that Windows works with bigwig files, maybe that's also another way you might want to do it. But it can take a BAM file, and you'll see why that's useful in this case. And a G ranges of regions we want to get the average signal over. So this is TSS's. So we're going to want to extract the average, or sorry, all the TSS positions for our genome. So if we want to plot over them, we need to get this G ranges of those positions. And we're in HG19. Thankfully, we already covered this. We know the TXDB packages contain all the gene models for our model organisms. So here we have HG19, and we could just load the TXDB, H. sapiens, UCSC, HG19 known gene. We load that with library load, and then straight away available to us is this object available. The object, the same name as the library, and that object contains all the gene models for this particular genome build. Okay. So this is how we can do it for known genomes. We also know, if you just go back to our genomic features lectures, we know we can create these TXDB objects from any GTF, any GFF. Right? We just do TXDB from GTF, and it will read it in as if it was a pre-built object from Bioconductor. Okay, so you're not limited. For those of you who's working on non-model organisms, you're not limited or stuck. You can still use these methods. You just need to import a TXDB object yourself from a GTF. Once we've got that, anyway, either from their objects or you created it yourself, we can just run this genes function here on our TXDB object. You could do, I said transcription start sites, so I'm using genes, but you could use transcripts here. Just be aware you're likely to double up and get multiple transcripts for the same gene. So here we're going to use genes. We're going to apply that to our object, and we'll get back a G ranges, like so, of all our gene positions, chromosome, start position, end position of the gene, the strand it's on, and then the ID. So these are entrees IDs. So this is alpha globulin or whatever. We looked it up and discovered what it is. But these numbers are actually IDs for different genes. So we can use our knowledge of G ranges here to use the resize function. And if we apply the resize function to our G ranges, we can set it to be fix as start, right? And we can say width of 1. Okay, resize is quite clever. Given a negative and a positive gene, right? We can see that here. On the negative, this is the TSS. On the positive, here is the TSS. And resize is clever enough that it's looked at the strand and it said, if you said start and you're on the negative strand, I actually meant the end, right? It's because genes are working that way. They're transcribing right to left in the genome. And if it was on the positive strand, clever enough that it took the start. Okay, so resize is nice and it's done a little bit of the work for us. We don't have to worry, wait, if it's negative, this is the actual start site. If it's positive, it's that. Okay. So that's done the work for us. We need to do just a little bit of fixing here. I'm going to take the main chromosomes. Remember, we just looked at the main chromosomes earlier on when we did the alignment. And I'm going to use a bit of a match. So I'm going to match the chromosome names across the TSS locations. I'm matching this column to the main chromosome names. And this is going to give me a position, and I can use that just to pull out just the TSS locations where I had, this TSS locations of genes which are in the main chromosomes, right? There may be genes which are on alternate chromosomes, and we don't want to use these. Then just fix the seq levels. We know seq levels are, all the potential chromosome names here. If I've removed some TSSs which are on a alternative chromosome and we don't want to use, I need to fix the seq levels as well. Okay. Given that we've done this and we have our TSS in a format where we just have genes represented in those chromosomes, we can now just pass this straight to Soggy. Okay, so Soggy's main workhorse function is region plot. And it's just going to take our BAM file and produce this meta plot we wanted. We just need to, at very basics, supply the test ranges, G ranges that we want to plot over. Okay. So we've got the load Soggy, got our BAM file here. And this is one of the ones you could download if you just want to follow along, and we'll get our plot. What's really nice and particularly useful about Soggy is that it has slightly better ways of dealing with BAM files. We don't have to pre-filter to select down to particular fragment lengths. Right? Remembering that we had these fragment bins that we know that anything less than 100 we consider to be nucleosome free. Anything from 180 to 240 was mononucleosome, and then 300 and something to something was trinucleosome. Okay, so often we would have to separate out our bigwigs, separate out, make a bigwig, or just separate out our BAMs up front. It's really nice. It's because this program, this package was written for ATACseq, or with that in mind. We also had these issues with having to have minimum fragment length, maximum fragment length. So it all applied nicely to ATACseq 2. So what we're going to do here is use our region plot. Going to give it our BAM file, give it our ranges. We're going to change something here to say style is point. And that's because we want to be focused on the center of these TSS locations. Just like here, we want it to be, this is our point of interest, and then we'll expand around our TSSs to look a little upstream, a little downstream. By default, I think it's, I guess, 1,500 downstream, upstream. So we set this, we tell it the format is BAM because it can be a bigwig too. We have to tell it it's paired. So you can give it the BAM. It's not going to check whether it was paired or not, right? So even if you gave it perfectly valid paired data, it will ignore it by default. We have to tell it it's paired. And then we say we want the minimum fragment lengths to include in our plot to be 0. Don't include any reads coming from fragment lengths or from fragments greater than 100. Okay, so this is going to be our nuke free. And then just to make the plot a little prettier, we can either reduce these down. A lot of people will reduce your fragment to the point, so you can see the center of where your fragment is, but we're going to make this 50. It just makes for a slightly prettier plot. Okay, so now we have this object, our nuke free. This is a, I think it's a check profile object. We can just pass that to plot region. So region plot being the thing which generates the data. Plot region being the thing which actually makes the plot. Just pass that to plot region. And it's going to give us around the center of our point, which is the TSS. We're going to see lots of signal, a little bit of a bias just before the TSS, and then less signal here. Do you see a little bump here? Right, this might be the +1 nucleosome. It might be because something isn't quite great about the data. But this is what we would like to see. Lots of signal upstream of the TSS, just upstream of the TSS. Right? Not much evidence of any +1 nucleosome signal. We can create the plot for the mononucleosome signal by just adjusting our fragment length and max fragment length to those which expected for those types of nucleosome fragments. So green leaf said 180 to 240, so we'll follow him there. And the only thing we need to change is 180 as my minimum fragment length, and then we get rid of all these mononucleosome, sorry, nuke-free signal. We get our max fragment length now to be 240, and then just from experience, I know it's force fragment length. I know if I center at around 80, we'll get a nice smooth plot. If I made this too small, my forced fragment length, I would get too very bitty. It would be very, very noisy. If I made my fragment length too big, then I just don't get the resolution. Okay, so then we look at this and we can see now, again, this isn't perfect. We'll check out Christina Leslie's and the exercises, and hers really is perfect. But what we do see here is this presence of mononucleosome signal over the expected +1 peak, right? So we see this really strong peak here, and this is our +1 nucleosome, +2, +3, +4, +5, maybe. And then also some upstream as well. We may be getting a bit of nucleosome. It's not a perfect separation here. I mean, ATACseq is not perfect. We may get a little bit of nuke-free signal here too, but this looks like we would expect, and we would be confident in this data. Okay, so I'm using this as a QC. Often people will take this and also compare signal over subsets of genes, subsets of TSSs. This we're not going to cover in this session, but we do have an exercise. We do have a course on this separate from everything else. It's on our page. I think Wei might talk about this on Friday. So a common goal in ATACseq is to identify regions which are either nucleosome free, so there's transcription factors are binding, transcription machinery is active. So the Greenleaf paper would say that this is less than 100 base pairs. So we might want to subset our data to just those fragments less than 100 base pairs. If we wanted to be sure we're talking about nucleosome free. Okay. Actually, I think the standards, like with bioinformatics, change over time. And if you can go look at the ENCODE standards of this, they've really moved the bar, what they would consider, how they would handle this data. And I can see the point is that really all this data is open chromatin. Even if it's nucleosome data, it's still nucleosomes from around open regions. We are throwing away a lot of reads if we just concentrate on the totally nucleosome-free region. So actually, we could just concentrate on open chromatin. And in that case, we would just use all the reads which were properly paired, and we wouldn't worry about any of this fragment length, insert size filtering. Okay, I recommend this one. Okay, this would be a new standard. For this workshop, though, we're just going to concentrate, and it's slightly to do with it just saves us time. We have smaller files. We're going to concentrate on the nucleosome-free portions of the data. Okay. There are many read methods available to call nucleosome-free regions in ATACseq data. There are those which are specific for ATACseq, but you go through the papers, you go through the what people are publishing. Really, people are adapting standard peak callers because they are well understood, and they have an incredible user base. Right? Once you start knowing how a peak caller works, and you've seen it and you run it on data, it becomes part of your quality control in itself. So things like Homer, which we're not going to cover, but Max2, which we will cover today, these have become very popular peak callers, not just for ChIP-seq and epigenetic marks, but now for ATACseq. And I think ENCODE uses Max2 in their peak calling. There is no package for Max2 yet. As we keep mentioning and teasing, for about a year, he's been developing it. I suspect in this release of Bioconductor, he's going to submit it. Maybe people in my team know he's already submitted it, because you can actually see. But it is possible to install it on Mac and Linux using Anaconda. Right? I don't think there's only I say unfortunately there's not a Windows implementation. I think Matt's checked this, but maybe you guys will find some Condor version of this. But certainly for the course, the one we're going to use, there is Mac, there is Linux available. Anaconda obviously has this huge collection of packages. It makes it very easy to install. And we're going to take advantage of Matt's package, Herper, which will allow us to just install this in a few lines, take advantage of it in a self-contained installation directory. We're not going to mess up everything else we have installed on our computer. And then we can either delete it or forget about it at the end, right? So we're gonna, like Herper is Bioconductor, so BioC Manager, install Herper, library Herper. Like I said at the beginning, Herper is new, not super, I think it was last release. So if you're not working on the last release of Bioconductor, if you're not in the latest R, you won't be able to get access to Herper. If that is a big problem, we can probably make available a version for you to get it to work. But I think ideally you would be on the latest version of Herper. So once you've got Herper installed, we have this very simple function, install Condor tools. And what we're going to do is install Max2 from Anaconda. And we're going to put it inside an environment on our computer called env ATACseq analysis. Okay. We can specify, particular other functions to say use this version of Python or Anaconda already on my machine, etc. But this will work for you, and it will just install it within the R location for Conda environments. And once we've got this working or installed, it should give us back, I think it just hasn't printed to screen, the paths to where this is. Okay, so this should take care of installing this, at least on Macs and Linux, in its own self-contained environment. And we can see later on how we're actually going to use it. I did mention this, but there are two additional arguments for this. Path to Miniconda, which is going to mean you can actually set not to use the default location. You can actually install this anywhere you want on your machine. Default location is pretty good. Update env. If you want to include new tools in your environment, you're going to have to say update env equals true. Okay, so given that we managed to get Herper installed, and that can get our external program in a place that we can take advantage of it and use it, we need to do then some peak calling. Okay, so for single-end peak calling, single-end sequencing, and like I said at the beginning, a lot of people do paired end for ATACseq, but for single-end, we don't know how long our fragments are. Okay, we can predict it. We can see maybe cross-correlation. We can get an idea, but we know we're going to have a different distributions of fragment lengths, right? So we don't have this advantage of being able to separate our data into long, short fragments. To identify open regions, so globally open regions in using Max2 for ATACseq, we're going to change some of our strategy. And this is really based on what ENCODE have suggested to do here. So one strategy is, and this is a strategy really just to call a broad region as open. Okay, one potential strategy is you shift your read back 100 and then extend the read 200 to the right. So you would move it back 100 to the left and then extend this to 200 to the right. This should, given the expected size of nucleosome-free fragments, provide a nice pile-up, which you can then call peaks on. Okay, so before we actually do the peak calling, we're slightly adjusting where our reads are in order to make really an estimate of where the center of our open signal is. I believe this -100 plus 200, it really is just to give us a broader region. Okay, give it a no model. Okay, I think we did this, maybe we didn't do this for ChIP-seq. By default, it will try and predict your fragment length. As we said earlier on, as I showed you, you will have distribution of fragment lengths. This is absolutely expected in ATACseq. When you do ChIP-seq, really you want all your fragment length, you want to pile up or you want fragments to be kind of centered on a mean fragment length, distribution around it. So it's very different for ChIP-seq. Okay, so we have to turn off any prediction of the model, and we tell it, rather than try and predict what your fragment length is, just take the reads, move it back 100, and then extend them 200. we can run this inside max2 inside R, now using this with Condor env function from Herper. Okay. So once we installed this Condor environment, it really is an environment, so it has other features. It's not just self-contained software. We need to set some environmental variables. We need to tell it, use this library here. So with Condor env, it just takes care of all of that for you. And what it does is inside the function, temporarily mounts that environment, just allows all these environmental variables to be accessible to you. And then we can make our system call within here to here, right, to max2. So we know the command is max2, right? We've installed it through Condor, so it's available to us once we don't have to give it the path, it's available to us. And we give it the arguments, call peak, right, call peak, -t single n bam, no model, shift, external size, format, -g. This is genome, we're telling it. Okay. So very simple, it's going to capture down that, and that will run peak calling there. Another approach for peak calling was potentially to shift and extend to the center of what you would consider a nucleosome here, right? So given nucleosomes are wrapped in 147 base pair, we would shift it 37 and then extend that read 73, which really should give us reads shifted over where the middle of our nucleosome was. Okay, still have to do no model, still have to tell it -g my genome. And then we can just run that. We're only changing a few arguments, the shift and the extension size. And then this will then go off and call our centered nucleosome peaks. Okay, this method is what you would normally run for single end, and this will work well for you. If you're running paired end data, actually we can just run max in BAM PE format, so BAM paired end. Okay, so then we can just do max2 call peak, give it our paired end BAM file, -f BAM PE. We don't need to worry about model or shifting or extension size, because we're not going to try and make our read look like the fragment should have been. We actually have the full fragment, right? We have both ends of it, so we can just, we don't have to reconstruct anything. We just know. Tell it the out directory. I'm going to give it a name, because we're actually going to keep this. And then -g, give it the genome, HS, MM. So in our case here, we're just going to set this with our openregions.bam. So again, I said you could use any peak, any reads within this 2000 fragment length range, insert size range. So we're using that BAM. We're going to name it this. We're going to tell it its BAM PE format. We're going to give it an output directory, and we're going to tell it human again. And we just run our system command to do all this inside our with Condor environment. Right? And anything you wanted to run from Condor, you could do this and then just wrap it in this with Condor env. Okay, so given we got some peaks from this, right, we're going to produce this peak file here. We should get 3 main files. We're going to get name, whatever we named it, narrow peak. So back in our genome data format, and I'm sure Doug talked about it yesterday, we have this narrow peak format, which is really suitable for IGV. It was developed, I think, as part of the ENCODE project. It's BAM format plus additional columns to store score minus log 10 p-value. What's nice is that narrow peak format is also well supported in IGV. It will color your peak by how the strength of the probability. Okay, so darker peaks, more confident. You don't get that with a bed file when you import it to IGV. So we also get this XLS file coming out. This is super useful in IGV. It isn't an XLS file. It is, right now it isn't an XLS file. Anyway, they may update this in the new Macs. It is just a tab separated file. They've thinly disguised it as an XLS by just calling it .xls, because your computer's going to try and open it in Excel, right? If it thinks it's XLS, it will try and open it in Excel. It realizes it's tab separated, but for you, it's invisible. The difference between this and this is that this file also contains the additional information on how we called peaks. So if I've forgotten what my line was for calling peaks, I can actually pull it out from my peaks at XLS. It's really useful if you ever want to see how someone called peaks. As long as you got this file, you can just work it out. Final thing, which is really useful, especially for motif finding, is the summits.bed. So this is the same as our peaks here, except it's giving us the summit position where we had the most signal within that peak. Okay, so we can see the shape of the peak, and this is probably the point where the center of where, say, a transcription factor was binding, and that's where we want to go search for the motif associated to that transcription factor. Okay, so we can do a little bit more QC now with our data. Often, a few things we want to check, and I think we already did things like duplicates. We're going to do that again here. Signal distribution. Where is your signal landing? We hope it lands in TSSs. We hope it lands in these peaks we've called. So we can assess all that, and we're going to borrow a little bit of functionality from ChipQC to give us an idea of reads and peaks, duplication rate, what are low quality reads. We did that manually earlier on. And I mentioned blacklist earlier on. We're going to try and see how many of our reads are landing in known blacklists. So to do this, we need to load, first download a blacklist, but we need to load ChipQC. I don't know why I'm loading DT. This is the data tables library. It's actually a library to make tables in rtrack layer, dplyr, and tidyr. So loading a few things now. We're going to import first our blacklist. So I think if you went here, this is blacklist. This is encode. They have blacklists for various data types here. This is GRC838, but you have HT19, MM10. So I've already downloaded that, and then I'm just going to import that blacklist. And I'm also going to import the peaks we've just called from Max2. And then with that, I'm going to pass to our ChipQC, and hopefully we saw this in the ChipSeq too, to the ChipQC function, ChipQC sample. Just give it our BAM file. Okay. Give it the chromosome we want to work on here, just chromosome 20. Give it our blacklist. It has built-in annotation, and it's going to use the TXDB objects we already use. So I can just say HG19 here. Importantly, I'm going to give it the peaks, because then it's going to give me things like reads and peaks as a statistic. Okay, so already this should, we run this. It'll take a little bit of time, especially on big files. It was written a few years ago. But once it's finished, it should have, first off, some very useful metrics on things like the proportion of reads, or the percentage of reads and peaks, and the percentage of reads in blacklist. So with our object returned, this is the, just off the side here, QC res is our ChipQC object. We can just apply QC metrics to that, and it gives us back a table. And it's actually only one sample, so it's just a one row table containing the ribble, the RIP, for instance. So this is quite low for reads in blacklist, but not bad for reads and peaks. 12% of your signal has gone into the reads and peaks. That's not too bad. Again, ENCODE has standards, so you can always compare yourself versus the standards in ENCODE. We also might want to pull out duplicates. This isn't quite the same as fragment duplicates, because we're just looking at individual reads. But I can do flag tag counts, and that will give me my flag counts. These flags are things like properly paired, mapped. We saw this when we tried to do some filtering. We used scan BAM flag earlier on. And then I'm just going to extract from this the duplicates by ChipQC, because ChipQC checks duplication rate itself. I divide that by the total mapped, and I have around 22% duplicates. So I think we had around 26% when we looked at the fragments, right? So it's a little less here. This is just because they do it slightly different ways. Mentioned this a few times, and I think Doug laid this home last session. So artifacts from sequencing and imperfect genome builds can confound our results. Okay. These artifacts have been collated for major genomes, and the one I showed you there, ENCODE, probably isn't the only source of blacklists. I think we discussed them in more detail here. But this is such a great example here, but this is for ChIP-seq signal. But you can see no matter what you sequenced here, you just saw all this signal piling up. And it really doesn't matter if it was an input sample, it's not going to matter if it's ATACseq or ChIP-seq. You can see these areas of just huge amounts of signal, thousands of reads tall. But these have been seen for a long time. So people have collated them. They've looked across lots and lots of samples for the different genomes. And using thresholds and cutoffs, they've decided on what we would say are consistently high artifact regions in genomes. So for sure, removing your blacklists before you ever look at your data is a bad idea. You want to be able to see how much of your signal really went into a blacklist in order to know what your background was. Otherwise, it's all foreground and your data looked great, but maybe I didn't get many reads. You really always want to look at your blacklist. The issue is that blacklists can confound some analysis. And in particular, they confounded this thing we totally ignored, which is this no model here, right? So when you tried to build a model in Max2 with blacklists, it just made it wrong. It was giving you very poor models, and Max2 itself built in mechanisms to get around this. But for sure, you want to eyeball and see how much signal you have in your blacklists, and then perhaps remove it before you start to do your downstream analysis, before you start to do real functional interrogation of your data. So in this case, we would still have some peak calls from blacklisted regions. We want to remove them at this point. So I'm going to turn my peak calls into a G ranges. It's because ChIP-QC is just this object we got out of ChIP-QC. Oop, where did I do that? This object here is built on a G ranges. So we can just turn it into a G ranges. Going to subset it to just those peaks on chromosome 20, because that's the only place we were interested in. And then I haven't printed it, but we could get some metrics on this. But most importantly, I take these peak calls and I just use my standard G ranges overlapping technology to say, give me all the peak calls which overlap the blacklist. I put the exclamation mark there. So I just keep peak calls which didn't contain any blacklist information. Okay. So now we have gone through FastQ, we've aligned it, and we've assessed some of the data post-alignment QC. We called our peaks and we've removed the blacklisted region, but now we actually want to do some annotation of these peaks, these open regions. We want to say, okay, well, this open region is near this gene. This gene perhaps controls immune response. You know, that means I'm seeing a lot of open regions around immune response genes. That might be something very interesting. Okay, but we want to annotate them to genes, and then from genes, we might be able to annotate them to the gene's functions. Okay. So one of the simplest ways of annotating a peak, an open region to the gene it's most likely to regulate is just to either look for the closest gene. So I say this open region regulates this gene right next to it, or this gene within 2000 base pairs. Or you can have things like a window. You can say any open regions within 2000 base pair window around my TSS, they're likely to regulate it. So it doesn't matter if it was nearest to my gene, it means this open region might be able to regulate multiple genes. Okay, but these are simple ways of doing it. You're either close based on a window, or you're the closest based on a certain distance. And usually it wouldn't just be closest, it's closest, but maybe not 100,000 kb away or 100 kb away. So there are lots of ways to do this in Bioconductor. Probably one of the easiest comes from ChipSeeker package. There's also a ChipPeak Anno package, which I really like. It's just not quite so easy to use, I guess. But what's the original? So we're going to take our TXDB object, TXDB H. sapiens UCSC H19 known gene. These are our gene models, and I said they're still important to us for this. And because ChipSeeker fits in with the whole bioconductor world, it accepts these gene models, and it's going to use them to annotate our peak calls to the nearest gene it lands in. Okay, so we're going to load our library, load our gene models again, and then I just use annotate peak. I give it our G ranges. I give it the just loaded object here, and it goes off and it goes and annotates this, finds the nearest features, distance, etc. As soon as I have that back, I can start to do some useful things with it. I think I don't actually have, it's a shame. If you printed max calls anno to screen, it gives you an overview. So it prints out an overview. If you want to get a plot of the overview, you can do things like plot anno pi, just give it our results, right? And it will make a nice annotation plot here. So you have promoter, 40% of our peaks landed in a promoter. That makes sense. 25% were distal intergenic. 0.17% were in our first exon. With this information, I think Doug goes more into this on ChipSeek. He's probably already been through this. With this information, we can actually extract a G ranges containing the distance to the TSSs, what that closest gene was. We can extract all that information by just doing as .g ranges on our max calls anno, and it will give us a G ranges, so like we had as in the bed file, but now with additional annotation here, our kind of annotation class, right? That's what got displayed here. The gene, chromosome start, end, length, gene strand, what that gene ID is. This is entrees ID, transcript ID, and then finally the distance to the TSS. So this is very close to the TSS. We might want to then subset our peaks, in this case, just to have peaks where they were landing actually really directly on a TSS. So we're going to take the absolute distance to the TSS. It has to be less than 500. And this will just give us open regions in the TSSs of genes in the HG19 gene model build. Okay, how are we doing for time? So once we have our peaks of interest, you know, either our regions here or those which are subset down to our TSS, we then want to do the next step. We know what genes they're associated to, but we want to know, are we associated to, say, more genes in immune response or in liver development than we expect by chance, right? Are we actually enriched for this? So one way would be to take the genes which we identified in ChipSeeker as having this open regions and test their functional enrichment using things like GoSeq. So I think this is exactly what Doug did yesterday. Just took those and you can apply this to GoSeq. Another approach would be to use the R-Great package or the Great. So I think both of these probably link. Brilliant. Yeah. So this is exactly what I said. This is looking for TSSs, and then we can do gene ontology on that. Or we have the Great one where you can use, oh, maybe that got jumped around a bit. Here you go. You can use Great. I think we had that in the ChipSeek course yesterday. Cool. Okay. Here, I think we're going to use great, are we? Yes. One thing to remember is our great will limit our queries. Actually, it's not so bad. There's only 14 of us in class. If we had like a class of 70, it starts to become a problem because I think they limit it on the IP of the university and not necessarily our personal IPs. So all of us doing this queries at the same time can cause a real trouble. We can actually get around this. There is like a function there, an argument they allow us to use, which will allow us to query more often. I guess by default, they turn it off so that we just don't savagely destroy their server. So we can submit peak calls to great, and I say submit. It's going to send this off to their server to actually run it. First, we need to load the library. Library are great. Tells us we are in version 4, which is great because we're now using HG38. Or we could use HG38, but you can, if you wanted to, specify what version you want to use. I think Doug went into this yesterday. If you wanted to use some of the functionality in old versions, you just have to specify. So here we're going to submit our great job, give it our peak calls, our G ranges. We're going to tell it we're in an HG19 genome, and it tells us the available categories that we can look at. And in this case, we have functional enrichment for Go phenotype. I guess this is things like mammalian phenotype ontology, and then genes. Okay, so if we want to dig into here, say the Go category, we then need to use this Git enrichment tables on our great job. And we tell it, I want the category Go. Then it brings me back, in this case, a list. And each one of those lists is a table of functional enrichment. Okay, so we have our great result. I have 3 elements in my list. I have 1 for Go molecular function, 1 for Go biological process, and 1 for Go cellular component. Okay, if I wanted to get like any list, I wanted to actually extract the table to do with this, I can just do double square brackets. I put the name of the element I want, and here I'm just going to look at the first 4 rows, and I can see I've got my Go for meiosis, monocyte, CD24, neurochrome, cell differentiation, and then my p-values here and a little bit off the page. Okay, so very easy to go from functional annotation from your G ranges, so you can start to see what genes are right next to your peaks of interest, and then to take that then integrate using R rather than having to copy and paste on the website, I think is also really straightforward. So I think we have 2 more sections left for this session. What I wanted to cover in the first bit, and I think we've already done this for ChIP-seq and we've already done it for the RNA-seq, was just how we can do a little bit of differential ATACseq analysis. And I think especially because of ATACseq, we don't worry about differences in antibody and efficiency. A lot of people do differential ATACseq analysis. And the idea that you could see changes in enhancer occupancy, enhancer openness, I think this really appeals to people. And I can tell you it works very well. If you have multiple groups and you have enough replicates, you can really pull out some very clear, functionally meaningful differential attack sites. So we're going to step through this, but it is very similar to what we would do in ChIP-seq. So my old boss, when I was back in Cambridge, was the author of diffbind. So diffbind is a fantastic tool to use. He's also my co-author in ChIP-QC. I don't think we're going to cover diffbind here. And the reason is, I think we have been through every single step. Diffbind is a workflow tool. It will handle a lot of it for you. But if you have been in classes with us, you should know exactly what diffbind is doing. There is one thing I didn't quite like in diffbind, and that is the way it defines its consensus set. But I think you can now jig this in diffbind yourself. So we're going to go and show you all the details. But for this and for the ChIP-seq, if you're interested, you should go look at diffbind as well. And diffbind starts at bands and peaks and does all the counting and everything for you. I feel personally it's nice to have a little bit more control. first thing we're going to do, and I actually deliberately, this is what I was hanging around at the beginning of the class to do, just to show us this. First thing we need to do is define a set of non-redundant peaks that we're going to do our counting and differential on. Okay. We want our non-redundant peaks to be in at least 2 samples, because that gives us here, I think, a good chance. We have 2 replicates. It gives us a good chance of it being in 2 replicates. Okay, you couldn't be more particular and say it needs to be in at least half of my replicates, but as you'll see, it's very easy to control this yourself, depending on how confident you want to be. So what we're going to do is take peak calls from all our samples. So every sample we've done peak calls, we're going to go grab the peak, combine it into one long G ranges, and then create this non-redundant peak set. Okay, so what we do is lapply. This is our directory of peak files. We're then going to, this is the one you can download, but also it was in our data directory, narrow peak. So we're going to do lapply for all the peak files. Here we're going to use git granges. This is a chipqc function, but you could use import from our track layer. So we're going to basically read in all our peaks, and then we're going to turn this list, because lapply, list of peaks into a granges list, because granges lists are a little cleverer than standard lists, and they allow us to unlist a granges list into a one long granges, and then we're going to reduce it to basically create this non-redundant peak set. Okay. Once we have our non-redundant peak set, we don't know then how many times or where peaks came from. We just reduced it, right? Where this peak came from. So then we loop through our original peaks, the G ranges of our original peaks, and we just give a matrix. We're just going to create a matrix down here of when a sample, when that peak occurred in a particular sample. Once we have that matrix, so, you know, I go through every peak at a time. I check for that peak set, where are the overlaps with our now reduced peak set? And once I have this all for every sample, I put it in, make it into a bind it into a matrix, give it some decent names, and then I attach it to my metadata columns from my reduced peak set. So actually I did the same. This is quite a lot happening on one slide, so I just wanted to show you. This is even worse because I've done it in one line, but we can see actually what's happening here. So the first thing to do is our dir, so we can list all the here narrow peak files, 1, 2, 3, 4, 5, 6. So this is where we have decided using max2, we have a peak, we have an open region. So, you know, in order to count and assess, we need to have something common for all our samples. So that's what we're going to do here. I can do this lapply, and actually I was going to read in all the samples. Here I'm not using chipqc, I'm just using our track layer import. Okay, and then I get my samples. Hang on a second. If anyone's here, I might not be sharing my screen. Am I sharing my screen? Stop share. Sorry, I'm just going to make sure I'm sharing my right screen. Okay, so in this case here, first thing is we can just list all the peaks we have. Next thing is we can lapply over those peaks and then import them using our track layer here. Could do chipqc. This gives us a list of G ranges. The real painful thing is that with a list of G ranges, if I do unlist, it just gives me back the G ranges again. So what we need to do is turn that into a G ranges list. Right, and once I have it as a G ranges list and unlist it, I get back a G ranges, right? So it knows, unlist knows how to deal with a G ranges list differently. It knows that I want them now in one table. Just doing that gives me 175,000 peaks, right? And these are peaks which maybe overlap. And then I can do our reduce on this to give me redundant, non-redundant, non-overlapping peaks. And we can go from our 1,759 to our only 89,000 peaks, which we have in the end. Right. So I want to show you this, what this looks like in IGV2. So here I have our reduced peaks. So what we wanted to do is go from a peak call in every sample here, right? And then I've reduced this into just one peak, which represents any of these peaks, right? So there isn't, if I flattened all these peaks together, I would get this peak. Similarly, I got this peak here. The next stage we do is actually to filter these for only peaks here, which occurred in at least 2 samples. So this peak is totally going to get filtered. It only occurs in this sample, right? And it's probably just a small background bit of signal. So we're happy to throw that away, but we want to keep these ones because maybe you have more signal in the liver than here, and we want to be able to count and assess. Okay, I should be sharing all my screens, so I can just hop back. Okay, so that's just to explain a little bit more about what's going on here. We're going from six different peak calls to just reducing this into one set of non-redundant peaks. And we can see that here. If we look at this, what we get is our now non-redundant collapsed region. And then I have a true or false matrix, and it's telling me, nope, this wasn't in this sample, it wasn't in that sample, it wasn't in that sample, but actually it was in this kidney sample. Next thing to do before we do any downstream analysis is filter out the blacklist. So we did this earlier on, but I'm going to do it again. Import the blacklist. It's a different blacklist because we're in a different genome now. Import the blacklist and just get rid of anything which overlapped the blacklist. And I'm also going to get rid of anything which was on chromosome M. Right. So I've shown you that chromosome M just has this inflated amount of reads. I'm kind of worried that really we're not going to get any sensible answers there, so we can just throw this away now. And worry about dealing with that in a slightly more refined way maybe later on. Once we have that, we have our here now 89,654. I had a few more here because probably we filtered out some in the blacklist. Now I'm going to take advantage of these true and false to try and filter out peaks which didn't occur in more than one sample. So now we can identify the number of samples in which our non-redundant peaks occur by just using the row sums on this occurrence matrix, right? On this matrix here. And if I just extract it from our metadata columns, turn it into a data frame, run row sums, I can get the number of times a peak actually occurred. And here I'm just going to pick peaks which occurred at least twice. Okay. Again, you might want to be more refined here. This is going to work well in this situation. You can be very refined. You could say, I want at least 5 times in this group, twice in this group. But in this case, this works well. Okay, it's going to clear out peaks which just occurred in one sample. So probably background noise peaks. Oh, this is also the diffbind default. So this is the same as what diffbind will do. Now we have a set of regions we want to count over, right? Our consensus, NR to count. We can then go and actually use this in our summarize overlaps function that we saw back in Bioconductor, and I'm sure RNA-seq and ChIP-seq. We just need to set or change one thing, right? So we can get our BAMs to count here. So we have our list of BAMs. The one thing we need to change is we need to change from single end, by default will be true. We change that to single end equals false. Now it's going to properly count while dealing with the fact that we have 2 reads belonging to the same fragment. Right? We don't necessarily want to count these 2 reads twice, right? Because they actually belong to the same fragment. So we run that, and then we'll just give this some better col names. I know the order, so it was hindbrain 1, hindbrain 2, kidney 1, kidney 2, liver 1, liver 2. So with our counts and fragments in our open regions, we can now construct our DSeq2 object. Okay, so here we're going to do, actually, we could have done this. Surprised we did it that way. Couldn't we just create it a little easier? But this will work. So I'm going to load in, here I had the counts already pre-created. So I'm going to load my counts. I'm going to give us, create our metadata information. So I've got some group information here, hindbrain, kidney, liver, matching the order here. And then I'm going to create my data frame where I have my group information. So that's this information here. I need my row names to match my column names. So I just set row names to be column names. I know the order. And then I can use this DSeq dataset from matrix. And I set my assay to be my counts. So I'm going to take out the assay. This is the table of counts from here. Going to give it my column, my metadata, right here. This is actually the cold data slot. I'm going to give it my row ranges, right? Because I want to be able to use row ranges later on in my filtering. So I'm going to give it the row ranges from my counts. So what I counted over, and I'm finally going to tell it I'm interested in testing over group, hindbrain, kidney, liver. Once I've built this DSeq dataset from matrix using count data, metadata, group information, this isn't necessary, but useful for us, our row ranges, what we counted over. And this will match per row, the rows here, and then our group information, what do we actually want to test over. Once I have all this, I pass my DSeq object to DSeq function. And as we saw, this big workhorse function, it's going to go find out the differences in library sizes. It's going to try and estimate global dispersions per peak dispersions and try and fit them to the global. And eventually it's going to give us an object with everything we need to start doing some differential testing. What we want to do here then is test, for instance, kidney versus hindbrain. We can just do results. We give it our DSeq object. I tell it I want to test over group. We set that here and also here. It's called group. And I'm going to say kidney minus hindbrain. So it's going to be kidney over hindbrain. And importantly, and different to RNA-seq, we also set the format to be G-ranges. Okay, so typically it's going to give you a data frame, which would be great. But we did add row range information here. So we should be able to get a G-ranges, and we did just that. And now I have my G-ranges object with important information like log fold change, p-value, p-adjusted off the side of the screen. But I have it in a format I can still overlap with TSSs, etc. Okay, so that's exactly what we're going to do. We're going to overlap it with our promoters. Yeah, so we're going to take our TXDB mass musculus UCSEMM10. From that, we will use their promoters function to extract plus or minus 500 around the start of a TXDB, start of a gene. And then going to do some little bit of filtering on my result from here. Okay, so I'm going to filter by those which were p-adjusted. Yeah, so those which had a score for p-adjusted, and that score was less than 0.05, and they overlapped with the promoter regions. And then I'm just going to use a function from here, track tables, to write this out and make a little report. Do we have that? Right. So I think we showed you a little bit of track tables earlier on. This is the make bed fart table, comes from that. It's just going to create a little HTML page which has all that information we just created, but really nicely as it allows us, hopefully I can click here. Oh, it should take us to that position in IGV. I think I've got some permissions issues on my computer. Okay. But the most important thing is, rather than doing what we normally do with with results from DSeq2, we can actually maintain the G-ranges. And this means that we can still work within that framework, use these G-ranges in Soggy to make average plots over regions which went up or down. It's a really nice feature there of DSeq. Okay. Following that, we might want to do just one last thing, I hope, and that is to say annotate our peak. Okay, so we just saw ChipSeeker earlier on. Here we're going to use our G ranges, right? Because it's a G ranges straight out of DSeq 2. Going to associate it to our TXDB musculus gene models. I'm going to say verbose equals false. Oh, no, I did eventually jump to that position from my table. I think there's some permission issues. It doesn't like me trying to take over things from my browser. So I get my annotated, and then I can turn that into a data frame. And now I have peaks, which are differential. And I know what gene they're closest to. I know the distance. And I can write this out to file. And this is probably something I would start to interrogate myself. Okay, this gene looks like a good target. I know it. And it has a strong fold change, and I'm confident in the significance of that. With these regions, you can then you can either take these to great, but just to show you something different, we're going to use Cluster Profiler here. We'll just load this library, and I can give it here the gene IDs associated from this table. Right? So we had a gene ID table, my entrees gene IDs. I tell it the genome of interest, our org.mm.egdb package, tell it the ontology, biological processes. Quite often these programs will have a limit. You can set a limit for how big your gene set should be. So I don't want any gene sets bigger than this size. And then we can just get the results from this. Okay. So very easy using DSeq 2, not just to get the counts into it, but also to maintain this GRanges structure and start to use all our other functionality inside GRanges and programs which use GRanges. Okay, so the last thing I wanted to do, I don't think it's a few slides, but it's a very simple concept, is this idea, and it's more a visual thing, but I think in the future we might add some more on this, because some of these programs like Tobias seem to use this type of idea. And that is this plotting, not the actual sequence, which is aligned post the cutting, but is to actually extrapolate where the cut site must have been, right? Where this T5 transposase must have been. And then by looking at the pileup of cut sites, we're kind of going to see the inverse. So we should see cut sites pile up around nucleosomes, or even cut sites pile up around transcription factor binding sites. And we're going to show you this now in these next few slides. Okay, so we don't necessarily need to actually have CHIP. We can use motifs as a proxy for where we think this particular transcription factor would bind. So I think in this example, we're going to use CTCF, because CTCF is a fantastically well-defined motif. CTCF is a very strong binding factor, so it's going to work well. And we're going to use, without knowing where CTCF has actually bound in the genome, we can just use the motif as a proxy in this case. So to get these cut sites from our bound file, we need to make some changes to the actual read. We're going to make our reads one base pair. We're going to shift them either +4 or -5, depending on the strand, in order to adjust for the distance between the actual T5 transposase cut site and what we actually sequence. Okay, we're going to use some Greenleaf data, because Greenleaf data was very, very deep. And we're going to take our CTCF motifs, we're going to find them, and use Soggy, which we saw at the beginning, to actually plot this cut sites now. So the first thing we need to do is actually find the CTCF positions across the genome. We need to know what a CTCF motif actually looks like in order to do this. So the motif DB package contains information on motifs from public databases like Jasper. So Jasper, I'm not sure if we've seen Jasper. But again, one of my old advisors used to be very, very involved with Jasper. Jasper is really just a collection of motifs, right? And we can see them on the right here, but the actual transcription factors here. So it's a very well curated source of motifs. So we can use this, the error information, to extract what a motif is like for CTCF. And to do that, we're going to use library motif DB. And we're just going to do query. Once we've loaded this, we just need to do query. We give it the motif DB object, which is loaded here. And we just give it a vector, in this case, just one motif that we want to query from this database. In this case, it's going to give us back CTCF, right? There's one thing I've queried for, but we have multiple versions of the CTCF motif, right? We have a Jasper early version, and a 2018 version has 3. 2016 they only had 2. Maybe in 14 they had 2. So there's options available to us here, and we can actually have a look at what's available. Hokomoko, this is the human mouse ones, and then, like I said, Jasper. Okay. So in order to extract, I think this has fallen off the end of the screen. I've actually extracted one, which I put into CD CTCF motif. But I'm sorry to say, it fell off the end of the screen. So we can just subset here, CTCF, double square brackets, and then we give it the motif we were interested in. And that then gave me this CTCF motif. If we want to visualize these motifs, like actually see what's inside them, we're going to cover motifs in more detail in the last session today. But the place to go is Jasper. I just had it open, right, but I've lost it again. Jasper. You know, because Jasper has this, what is a Seq logo on the right, and you can see this is really the proportion of times at a position in a motif we see a particular base. So this T always occurs at this position in this motif, this G as well. This occurs quite often, but sometimes it's a T, and this one much less so, T, C. If you want to, you can also make these plots inside R using this wonderful package, Seq Logo. And Seq Logo just accepts a PWM straight out of motif DB. So we didn't have to do any more work. We just subset it here, and then I just pass, once I've loaded the library, Seq Logo CTCF motif. And you can see it gives a display of the motif here. Okay, we're going to go a little bit more into what this plot really means later on. But you can see, well, what's clear is the bigger the size, the more frequent that base occurs at that position in the motif. This is 19 base pair motif. And in the middle, we're very confident in G, but there is a potential to have an A, a U, and a T. Here, you know, we're not so confident. Maybe we're most confident in T and C, and here we have no real confidence. Okay, the total height here, the information content, really is how great is our strongest confidence. Okay, so when we're not confident of anything, that base has very little information, and you'll be very small overall. When we are very confident in one base over all the others, the information content is high. What you can see, which is clear, CTCF is a complex motif. I showed you Jasper here. This is a really simple motif, right? CTCF is a very complex motif, and this is going to allow us to actually very confidently identify its positions. Okay, so we're going to go in more detail into this later on. But if we want to scan for PWMs for, you know, this pattern, this frequency pattern over a genome, we can just use, rather than match pattern, which we used earlier on in our courses, we're now going to use match PWM. Okay. So to work with match PWM, we just need to give it our PWM we just had here. Okay. Same thing we're using for our Seq logo. And then we give it a DNA string to test over. Okay. So here I'm extracting the DNA string for chromosome 20 from this HT19. Now, at the same time, we're just doing this match PWM, and we're going to get back a new object to us, but hopefully we can recognize a big part of it. This is a views object. which is basically an iRanges object with some sequence information. Okay. If we want to, we can actually just turn this back into a GRanges. We just need to do GRanges. We build it ourselves. We give it the chromosome 20. Right, that was the only bit of information missing here. We tell it ranges, my ranges. Right? So we extract these ranges proportion from here, and we're back to something which we can start to work with. Right. So now we have a GRanges for chromosome 20 of all the CTCF positions. To plot cut sites, we actually need to do a little bit of editing of the read. So we're only going to consider the 5' end of the read. I'm going to need to adjust this 5' end to account for the slight shift from where the actual T5 transposase cut. TN5. Yeah. So that means we're going to capture the 5' end. We're going to shift it depending on the strand, and then we can use that to then do the plotting. First things first, we'll read in our data. We have our BAM file. We'll do read GA alignment pairs, right? So we want them to be this GA alignment pairs object. And then I'm going to extract the first and the second read in the pair separately. So we have read 1, read 2. You can see read 1, read 2 here. I'm then going to do this to both of them one at a time, but I'm going to resize our reads on read 1, just like we did with GRanges. I'm going to turn it into a GRanges. I'm going to resize it so that the start of the read is 1 base pair. Right, so this means it's just going to give us the 5'. And then 4 reads either on the positive or the negative strand, I'm going to shift them a little differently. Right? If you're on the positive, I'll shift positive 4. If you're on the negative, I'll go back 5. And then I'll do that for my second read too, my other end of my fragment. And once I've got all my cut positions for all my ends of my fragments, depending on the strand, I put them all together into a long vector, a long GRanges, and then we have it here. Right, so this is all the cut positions now for this ATACseq data for chromosome 20. And you can see, because they're a cut site, they're going to be just 1 position. From any GRanges, as we said, we can make coverage, right? So we can turn this into an RLE list, which is equivalent to a bigwig, but internal for R. So here I'm just going to pass our cut sites from here to the coverage function. It's going to give me my coverage, cuts coverage RLE list. And then I'm going to subset that just to a chromosome 20. Hopefully we can recognize this. This is our RLE, part of our RLE list. And it's just the coverage, the number of cut sites across this particular chromosome. So what we really like about RLE list, or RLE, is that Soggy will also take RLE list. So it can accept BAM files. So you've already written it out, or it's external. It can accept bigwigs, right, because they're already produced. But if you want to edit the data internally, make your own RLE list, you can also use that. You just need to do cuts coverage here. So our RLE list object, change the format from BAM or bigwig now to RLE list. I'm going to do style point again because we want to center on the middle of these to compare, I think, was our motifs for CTCF. I want to center in the middle of them, so I'm going to use point, and I want to go 500 base pairs up or down. By default, 1,500. And now I can pass that to my plotting function. And you can see with very simple bit of coding, we can see we have cut sites really piling up around CTCF. CTCF is famous for producing great versions of these plots. If you were to do a nucleosome length plot, so a nucleosome signal plot, you would see the pile up of nucleosomes well positioned around a CTCF site. So it's a really good example. One thing I did have to change for our plot region is I set this thing here, outliers equals 0.001. This is just that occasionally you will have a point in your plot where you'll just see a huge amount of signal. And this is not necessarily a blacklist, but it's somewhere in your data, one of these TSSs, you have some artifact signal, right? We can remove points which are considered like huge outliers, and this is kind of default, and it will just produce a much cleaner plot for you. Okay, so that's a long session. We have some exercises here. Hopefully you can already get the data, so you can already start to do some plotting. I just wanted to show you this because this is really beautiful data. This is Christina Leslie's data. If you put this through the same thing we show in class, you see you get an even cleaner pileup of the data there. And then the +1, 2, 3, 4, 5, 6, 7 nucleosomes piling up as you move along. And again, this is only one chromosome I think we're looking at here. It would be even better. 43% reads and peaks rather than 20% we were seeing earlier on. So worth looking at the exercises for this one in particular, because it will give you a good idea about the standards which say MSK in the last few years have been able to produce for ATACseq. If, again, you want to skip ahead, you got your answers here too, and you can just run along and get it together. But I'm going to leave you open with that plot. Maybe I'll do it with the next exercises, just to show you how far they came in going from the first paper to now ridiculously 1, 2, 3, 4, 5, 6, 7, that you can really pick up huge resolution, great resolution. Okay, so I think that's the end of our session now. I will be back at 3 o'clock to run through our final session, which will be learning a little bit more about motifs, and how we handle motifs and different motif libraries in Bioconductor. Cool, I'll see you a little bit later. Knowing that we look at the distribution of our fragment lengths here, we have a high proportion of data which is less than a mononucleosome size, right? This is our free regions, our regions around transcription factors, such as this here. Okay, most of our data is coming from here. A little bit of our data is coming from the nucleosome. Another side of ATACseq we're not going to look at is actually studying how nucleosomes are positioned. We can use a bit of this information. And, I did a lot of this work in amylase data, where we'll look at, fragility of nucleosomes. We'll look at whether or not they're stable or fuzzy. But as I said, most of us will be using ATACseq data really to concentrate on this transcription factors. We want to know, given we found an open region, what is the transcription factor in that open region? Okay. This is the real advantage, this open regions around transcription factors or epigenetic machinery. This is the real advantage over RNA-seq. Right? Otherwise, I could just use RNA-seq to measure changes in gene expression, and I could do functional enrichment on that. ATACseq can be used as a proxy. By seeing more open signal around a gene, we can assume it's higher expressed. But what ATACseq offers us is this ability to really drill down and look at the motifs under these open regions and regions away from genes. If this region has shrunk or gained, as we did look at with liver, kidney, and hindbrain, if I have a region only in liver and not in hindbrain, I might want to look at the motifs underneath that and try and understand what transcription factors are acting in the liver, but not in the hindbrain. Okay. The difference, though, is, and this problem here is that ATACseq's given me all the open regions. I have no way of knowing what is underneath them just from the ATACseq data. Right? It's not like ChIP-seq, where we have a IP. So I have an antibody for a particular factor. When I do my ChIP-seq, I've got my DNA bound to proteins, and I use a factor to enrich for DNA bound to that protein, an antibody, sorry, to bring it down. Here, we're bringing down all open regions, and we have no knowledge whether or not a transcription factor is in them. Okay? ChIP-seq's targeted. ATACseq is any open regions. So we looked briefly that we could use, and we looked very early on, we could use things like match pattern, right? If we have a regular expression, maybe we want to look for a tartar in the front of genes, TATA, tartar box. Maybe we want to scan for something simple. At the end of last session, we saw that we can actually use this match PWM, and this is available. Have I got power? Yeah. This is available base R, right? We can use this straight out of Bioconductor. And it allowed us to scan a whole genome very quickly or sequences and find where the motif was within them. So that's great. And actually match to PWM was a big workhorse in Bioconductor for a long time. A few years ago, the same lab, which did ATACseq, like developed ATACseq, this one-stroke Greenleaf lab, I guess. Also in that lab, they started clearly wanting to investigate the same questions we all do. What motifs are under my ATACseq? How is motif affecting the accessibility across conditions? So they developed this wonderful package, Motif Matcher. And we're going to look a little bit about how we can use the motif databases, so not now gene model databases or sequence databases, motif model databases, and how we can use that in combination with Motif Matcher to scan our genomes in really efficient ways. So Bioconductor provides two major sources of motifs. We've already seen one, it's the Motif DB package. Now the other one, Jasper 2020, or the Jasper database packages, I think they have 2018, 2016. These are a little bit more up to date, and we're going to look at both of them, but we're going to be concentrating on this second one more. So the Motif DB package collects motif information from a wide range of sources, and it's going to store them in a DB object, just like our TXDB, just like our BS genome objects. But it's going to allow us to use this with other Bioconductor packages. So we can load the library MotifDB, and then if we just print MotifDB, type it into the console, just as we did for when we loaded TXDB, we then take the name of the package and take that object and type it into the console. It gives us information on the object itself, and it's saying, I have 10,000 motifs, telling you when they were produced, a little bit out of date, created from downloaded sources 2013, and then tells you some information about what's in there. Okay, I think we saw Hoko Moko earlier on. This is the human mouse particular motifs. Oh, and you got fly factor as well, that's good. If we look at this object we just got back, MotifDB. So typically we have things like, for the TXDB, it was a TXDB genomic features class. Here, this is a motif list. Okay, so this object we get back once we load is a motif list. And as always, it tells us it's from the MotifDB package. It is like a list. We can use some of these list functions. You know, it's in the name itself. So we can do things like length, and we'll get the total number of elements in this database. This is going to match this number here. We can also get the names, so I can see all the motifs in my database. Maybe I know that I'm looking for this particular ID, right? So I can get them names just like I would with a list, and I can list them too. I can show them too. We can also access information directly from our list using the standard list assessors. So what we would do normally, like a single square bracket here, will subset to a single instance of a motif list. So if we have a normal list and I do a square bracket 1, like here, it's going to give me back a list still just containing the element I've asked for, in this case 1. Same here, right? When I do MotifDB square brackets, just 1 square bracket 1, I get the same motif list object back, a MotifDB object, they call it here, but it is a motif list. But in this case, it's now just length 1. It's a little bit easier to see. With double square brackets, I can really dig down inside my motif database. And I'm going to get actually kind of nicely what I would want, which is my position probability matrix. Okay, so this now for this motif, which is whatever this is, MO1. Okay, I guess we could look that up in Jasper somewhere. We can go look that up in Jasper later. I can now actually get the values associated with this just by doing the double square brackets. Okay, let's draw that up. Position probability matrix is exactly what you might expect. It is working across the bases. The probability of this being a T, 0.97, so it's very likely a T. And then this is likely a T. This is likely an A, right? So this is just a very simple one. The probability for every position in the motif of it being a particular base. If I can prove this by just doing the col sums of this particular motif, and it will add up to 1. You can also get very usefully a data frame of all the motif metadata within our object by just using this new function, this new accessor, values. So if I do values on my MotifDB object, is going to give me back the bioconductor data frame. So rather than being data.frame, this is capital D, capital F. Acts just like a data frame, displays a little differently. And it's going to give us additional information on this particular motifs or the motifs of interest. So here we have the provider name, provider ID, the actual source, the protein ID type, the protein ID, the organism, the sequence name, its count, binding, whatever. Really usefully, we have gene symbol. So now I no longer need to worry what MO1 is. I know it's ABF1. I know this is AFT1. And although it's a little bit off the slide here, we also have the transcript family. And that's, June motif and a FOSS motif. They may be the same family of motifs, very similar, but they would have different, obviously, gene symbols associated to them. Okay, so we can extract from this. We can dig down to just one object, and this gives us a little bit of information here, but this object isn't really useful for us to work with, right? If we want to extract the underlying probability matrix, what we may use to scan our genome with, we can do this. Double square brackets. And if we want to get like data frame of information, including importantly, what is the actual transcription factor name? What is the name of this transcription factor associated to the motif? We can just extract it by doing values, get the data frame, and then get the gene symbol. And we could use it with this, maybe extract these two columns, and we can do merging and matching as needed. We can also query, and we saw this in our last session. I just didn't really go into it in too much detail because we know we're coming at here. We can also use the query function from inside the MotifDB. We can use query. We can give it our object, and we can tell it I want to query CTCF. And then I'll get back a motif list with all the instances where it's found a match with CTCF. Okay. So it searches through all these data frames to try and find out whether or not we have a match for CTCF. Okay, so actually we have lots of CTCFs. We have 19. Probably a lot of these are going to be the same motif, just collected in different sources. I mean, undoubtedly Jasper 2014, 16, 18, they're going to contain copies of the same motif. Okay, so it's not, there is some redundancy here at this point. And I think this is quite a common problem. People will do motif identification, and they will find both the Jasper 2008 and 16 version of the motif, and it just crowds your analysis, really. We can filter down a bit more. So we can actually be more specific. And again, if we say here now CTCF, H. sapiens and Jasper 2018, it's going to again go and scan through the gene, the organism, and the provider, data source, sorry. And it's going to check the columns. It's going to make sure we have matches for these, and they will bring back just a much more subset motif list now, just containing those from Jasper 2018, H. sapiens. And actually, we can see there's a little issue. It's pulled out CTCF, but also CTCFL, which is, I think, a slightly different transcription factor. Okay, so that's the MotifDB package, and actually a lot of manuals use the MotifDB package, right? It's built, Paul Shannon, he's also the author of IGVR, right? But it was built a long time ago, 8.5 years, and it probably isn't that often. Well, we know it's not updated often, right? Probably see somewhere here when he updated it. See, the package gets built, but the as we saw in our R session or in our code, you know, 2013 August, it was just the last time they bothered taking that, and it keeps on getting built. The package, but not necessarily updated. Okay. That said, you know, they do have Jasper 2018 in there, so. The Jasper package, I think, is probably the place to go. Jasper, for a long time, was very unsuccessful in their, it's a consortium of motifs. We can go here and look at it. So this is Jasper. They were very unsuccessful, I think, in really forming a nice consortium for a while. And there was equivalent called Transvac, which was just awful. Really tough, and they had some versions, some motifs you could download, and if you wanted to pay money, you could get slightly more. Luckily, Jasper, around 2014, they had a release, and they really did a great job. There's a big consortium behind it now. And now it's become a really good, trusted source of motifs. And in fact, the Jasper 2020, or at least the Bioconductor library, these are all written by Boris Leonard, and he was my old advisor. So we can trust in these packages. They're always going to give us something modern and robust, right? We're not going to be out of date, and it's in the name actually when it got run. So if we want to use this, we want to get advantage of all the motifs. I can tell you again, to stress how important it is to be cutting edge. We did a paper 2 years ago, and I used Jasper 2014 or 16, and it was missing the motif we needed. Right? I updated to Jasper 2018, and I had the motif. So we could see it. We could see we're pulling out what was clearly an ATF4 motif, but we weren't able to identify what it was using Jasper 2016. So it's always worth keeping up to date. In this case, we're going to use this one. Right, so once we've got it, we can just, I was on the page, I think, but you know, you can just Google that, just 2020, go to Bioconductor, Control F this, and you can install it using BioC Manager. Once we have this, like all these packages, we can just take the name of the object, the name of the package. We have an object the same name. And if I print this, so if I type this into R, we actually don't get this beautiful thing we got with TXDB. We don't get this nice thing we got with MotifDB, where it gave us, you know, this lovely output for when we just printed this to screen. It just tells us I'm a DB and this is my path. Okay, and that's because at the same time they were writing Jasper 2020 or Jasper 2018, I think they've done consistently a package every time there's been a release. The author of TFBS tools is in the same lab. This was Gertan, or he was in the same lab with Boris Leonard. And they've kind of split the functionality. So it's very, very sensibly. TFBS tools is going to be consistent, right? This is how we interact with these databases. Jasper is going to be updated every couple of years, right? So that's going to change, and we'll need to use Jasper 2022 next year. But TFBS tools is always the same. And we're going to show you also, there are places obviously to import your own motifs as well. So we'll come to that at the end. So Jasper package holds the information on the motifs and position probabilities, and it's TFBS tools, which has the functionality to really interact with them. Right? So it's a little bit like TXDB packages and genomic features, or BS genome packages and, you know, biostrings, right? On their own, they're kind of useless, but once you have the functionality to interact with them, they become invaluable. So 3 functions which we have available to us in TFBS tools, which are really useful, are get matrix set. So get a group of PWM matrices, get matrix by ID, and get a matrix by name. Sorry, I've written get matrix by ID twice. If you want to have a look really how these work, we can do this now together. We can just do a Control F. I'm going to load the package and make sure I've actually installed the package. TFBS tools, great. And get matrix by ID, question mark. You can see all the methods available to us. And I think it gives a good description somewhere here. Oh, get matrix set maybe. So now we have a really lovely description of all the different things we can use. And just like with most help pages, it is in there somewhere. You may just not find it on the first help page. You have to click through. But you can see here we got things like class, type, tax, all these things we can use for filtering when we start to do things like get matrix set. Okay. So the get matrix by ID and get matrix by name take the Jasper DB object. So what we had when we loaded this object here, and a Jasper ID or a transcription factor name based on ID and by name. And then we're able to pull us back a new object, which is a PFM matrix, point frequency matrix. So we can do that here. Once we've loaded TFBS tools, we can just do get matrix by name, give it our Jasper 2020 database object. And here I'm going to use Gatter. I look at the class, I get my PFM matrix from the TFBS tools package, right? It's not from Jasper package, it's from the TFBS tools package. Similarly, I could do exactly the same with Jasper and using this get matrix by ID. So I already told you, and we can see here again, sometimes, this isn't quite the case, but sometimes you will get multiple motifs for the same transcription factor. Different places have different ideas about what they consider the right PWM for a transcription factor. In that case, we can also use this get matrix by ID, and we can specify the exact transcription factor we want. So if we just step out to Jasper again, probably look here, CTCF. And we can see we have a few versions, but maybe if I didn't specify the species, I would have 2 CTCFs, one for Drosophila, one for Homo sapiens, but the ID here is unique. Right, so this is actually I'm not sure what type of ID this is. It's a matrix ID. I'm not sure where it comes from. Maybe this is actually a Jasper ID. So if we want to be specific, we can also use get matrix by ID, pull it out that way. List accessors aren't going to work with this, sadly, but we can retrieve the names from what we've pulled out by doing ID and then giving it the PFM object matrix we just retrieved. Right, so now with this Gatter map, however I pulled it out, I can still get the ID for it using ID in my PFM matrix, and I could take that back and go into Jasper and actually see what that was for. You search Jasper. There you go, Gatter, and that's what it looks like when you actually display it. So if we want to get actually hold of this PFM matrix, this point frequency matrix here, we can use either the matrix function or the as.matrix function to convert that PFM object into a standard matrix. So we actually did this as.matrix, I think, in one of our earlier classes, one of our earlier sections, workshops. Very simple, just matrix, give it this PFM object we just created here. My matrix, as.matrix, going to give me the same thing. And then I have now my positions along the top. So this is the first position, second, third, fourth, fifth, sixth, seventh, eighth. And then we have the frequency for every position. So here, out of all the ones we've assessed, out of all the sequences, 1,569 had an A, 1,490 had a T, 567, 700. Okay. So if I just stacked up all my sequences and I reviewed how often we have an A at that position, how often we have a T, a C, and a G. Okay. You can also use the Git matrix set function, which is probably more useful and what you will use really when you want to do a global motif analysis. So Git matrix set function is going to give us sets of motifs. It's going to be the same object back. And we can specify a list of options for the motifs we want to retrieve. Okay. So best way to see what options are available to us, again, is this Git matrix set. So if I just hop into R, I think this is what I had open. No, oops, double question mark. That'll do. And here you can see all the options. Collections, what can I specify for collections? What can I specify for all versions or species? In Latin, Homo sapiens, or NCBI taxa ID. So it's here that we can really see what options are available to me in order to do my filtering for this Git matrix set. And at the end, I always have this nice example that shows me that I create an options list. I then fill that list with things like the species I want, the type I want, whether I want all versions, and then obviously to know this, I just had to go back up here and I said something about species. Yeah, species. This needs to be either the Latin or the ID. So here they've chosen the actual ID, 9606. And this is the NCBI ID. Okay, so with a bit of help, looking at the help, we should be able to work out how to filter. In this case, I'm going to provide some filters that I want the core collection, so the main collection from Jasper, and I'm only interested in vertebrates in this case. So the taxa group is going to be vertebrates. And then I can just run my Git matrix set on my Jasper DB object, give it my options, these ones I've set here, and it's going to give me back another PFM list object, PFM matrix object. Okay, so that's how we can use Motif DB, but more importantly, Jasper, to go in and pull out individual motifs, but most likely motif sets in a bioconductor object we can use later on. So the Seq logo package we saw in the last section, it offers a simple and intuitive way of visualizing our base frequencies within our motifs using the Seq logo function. Seq logo plot package has one function, Seq logo. So it's a cool package. A simple Seq logo without accounting for differences in information content just shows on the left the probability, right? And we saw this when we did our PPM here, right? It's just going to take that matrix and use that to stack up how high a T would be, how high a C would be, a G or an A. In this case here on position 5, we can see it absolutely should be a C. So we'll have that, most of the probability, and then there's a little bit of probability here. Okay. So in order to create this PPM, we need to use a PPM in order to give to Seq logo. We saw earlier on we could just grab that, and that's why I just showed you straight from the Motif DB package, Motif DB package for CTCF. So that's exactly what we did earlier on. This thing here, this is what we want to extract from inside the Motif DB package. So to do that, we're just going to use Seq logo, and we're going to do query for CTCF. And then we saw that we could just use double square brackets to get that matrix straight out. So I'll just do that. And I'm going to pass it to the Seq logo from the Seq logo function. Here I'm just going to set, and we'll come to this in the next slide, IC, so information content scale is false. So we're just going to see probabilities, and this is exactly what we see. Right? This is our CTCF motif. It's capturing the most important information of where Bases are very particular. G here, A there, C's here. Often you'll see our Seq logos more like this, right? You won't see this crowded window of values. You'll actually see it like this. And it's just because this is just a much better way of really telling you what's important in your data. And what we have here is actually information content. So you can look up how information content works, but in reality, it's going to range from 0 to 2. If you have equal probabilities for your bases, you're going to score very low here. So if all your bases had the same probability of being at this position, you're going to have a very low score. It's going to have very low information. It's not telling you anything. If you had like 10 here, and this was a position where it was pretty much always G, then that's got a lot of information. It actually tells you great certainty that that should be a G. Right? This is how you're going to see most of your Seq logos. To get it, it's actually the default. You just do Seq logo, and we give it our PPM matrix, our probability matrix, straight from MotifDB. Cool. So we don't necessarily, I made a point about saying it's a bit out of date, MotifDB. So really we want to be able to do the same using TFBS tools in Jasper. But as we saw earlier on, it didn't give us a PPM. It actually gave us a PFM, point frequency matrix. And you can see it here. This isn't going to be able to display this, or we're not going to be able to display this directly in Seq logo. But we know that it's kind of easy to turn it into a PPM. All we need to do is take our matrix like this and divide each column by the sum of the column, right? And that will tell me the probability. So I can do that here. I take my matrix, and this is a nicer way to do it. Now I just divide it by the col sums of my matrix. So I'm dividing the columns by the sums of the columns. And then I get my PPM here. Looks plausible. I can then take that PPM and pass that straight to the Seq logo. And then I get my information content and my point probability matrix displayed. Okay. This is, I think, if we had it open, that's the same, right? So we can get it from we're going to get the same pattern, same logo as we did there. So we do want to do this from TFBS tools, and we don't want to convert it all ourselves every time. Clearly, we don't need to. TFBS tools have thought about this, and they actually have a new class called ICM class. Oh, sorry, ICM matrix, IC matrix. So information content matrix, this is what they like to call the PPM. So in order to convert our matrix, our matrices, our PFM list to something to display an ICM matrix class to display in Seq logo, we just need to do 2 ICM, give it our standard PPM, PFM, and it's going to convert that to an information class matrix, ICM. Now we can use Seq logo, but actually we're going to use a new version of it from TFBS tools. And this is why I'm doing the double colon here, is just to specify which package we're using. So in this case, it's still the Seq logo function. You would not notice if you installed it that you were using it from a different package. But this is the TFBS version, and it's going to give us exactly the same plot. Okay. No matter where we come from, we do it ourselves manually, then plot it, or we take it and let TFBS tools do it, and we'll plot it the same. I want to mention this package because a long time ago, we love ggplot, and we're a big fan of it. A long time ago, I saw a post by the ggplot author, Hadley Wickham, saying that, you know, he really hated these plots and he absolutely hopes that we can't do it. Actually, these plots are super useful. And someone has come along and written a library in ggplot to allow us to do these plots and also take advantage of all the wonderful GG-ness in theming, etc. So here we're just going to use ggseq logo. It's load the ggplot library because this will allow us to add a theme. And I just give it my matrix, right? So the original PFM here. And it will look at this matrix. It works out that it's a PFM, not a PPM, and it will divide by the col sums. And again, we just get the same. Okay. 3 ways to do the same thing. But what's nice and reassuring is that each of them are going to give us the same answer. So now we have access to the motifs we want, and we can filter this down to the correct subset of motifs really relevant to us. We now want to actually try and scan our regions of open or accessible signal and try and identify what motifs may be underneath these peaks. So this particular region which has gone up in hindbrain, maybe we have a particular transcription factor motif there, which relates to a factor expressed only in the brain. Okay. So we're going to use, and I mentioned this earlier on, this motif matcher package. Motif matcher is really just a wrapper for a C++ library, the mood C++ library. And this is why we wrote FastP, rFastP. It's because we saw it was possible just to use C++ libraries very easily inside R without having to leave R, use Herper. We can just actually wrap up the C++ directly. So in this case, this is what they've done. And it just means we have a few functions in R which allow us to pass it off to this really fast library to go match against all these peaks and all these motifs. Okay. This is going to make a massive difference because we have lots of peaks, we have lots of samples, and we need to do this fast if you want to be able to iterate over new ways of looking. If you want to try different things, you don't want to wait an hour, 2 hours for it to do the scanning. Another nice thing is Motif Matcher has a good way of defining a p-value cutoff for motifs. So we don't have to say, you know, it has to occur a certain amount of times. It's going to work this all out for us. So we can define we can, with it, identify confidence motifs across the genome or in our peaks or in our regions of interest. So first thing we're going to do is get a sensible set of motifs to scan over our mouse tissue ATACseq. So this is going to be hindbrain, liver, kidney data. So I'm going to do this by using the Git matrix set. I'm going to give it Jasper 2020, and I'm going to set my options list. It's empty at first, and then I create an element called tax group. I put in their vertebrates. Then I'm going to put collections. I'm going to want just the core. Finally, I recommend this. I'm going to say all versions. I'm going to say false. So I just want the last version of a particular motif. I don't want previous versions as they've got there. That's really useful for when you want to track. I used this motif 3 years ago. Oh, it's very cold. I used this 3 years ago. I want to know what that motif was, but in your analysis, you want to use the latest. With that, I can pass that options as my second argument to Git matrix set, give it the Jasper DB, and it's going to go get motifs to scan, which will be our PFM matrix, and that's going to be input for Motif Matcher. This is the nice thing. Motif Matcher clearly looked at TFBS tools, Jasper, and said, we want to be able to support the objects coming from there. So the main function, this big workhorse function for Motif Matcher is match motifs. As with other bioconductor functions, match motifs, and this is what makes it much more than just a wrapper for this C++ library. Motif match motifs can take other bioconductor objects, such as a BS genome object, such as a G-ranges, and summarized experiment objects. Can work with all these, and this should speed up our entire kind of analysis. Again, if you want to have full detail of help, you can do question mark match motifs, and that will show you all the things you can do. So we need some data to review. We have this ENCODE mouse tissue ATACseq. So this was our hindbrain, liver, and I guess haven't got enough space, but it's going to be kidney as well. Okay, so I'm going to load that from our counts. We would have done this earlier on. This is when we were doing our peak calling, our differential attack. So this is the same data. And that means we have 36,000 peaks and 6 samples. because we made a range summarized experiment, and I said this will be super useful, super useful to us, that means it actually contains in this slot row data. Maybe not. Row ranges, I guess, is off the side of the page. We have ranges still contained within our summarized experiment object. It is a range summarized experiment object. So we can just extract that by using row ranges, giving it our summarized experiment object, and then we have the G ranges which were counted over. Okay, so this is going to be in the same order as the matrix was. The rows will match these rows, and then we had column information, which would be the different samples. We know back in our first lectures, we learned a little bit that we can use things like GitSeq to extract sequences from BS genome objects using G ranges. So we can start to use this GitSeq now with one object type and another object type. So starting to cross across our packages in order to extract the sequence underneath our peaks here. So we did a little bit of this, I think, for ChIP-seq. We used some de novo there. So here we're going to load the BS genome for appropriate place, musculus. We're going to recenter our peaks to 100 base pairs using the resize function. And once we're resized, we can extract the sequence. So we're going to load this library. Going to resize our peaks. We're going to fix on the center of the peak. So the geometric center. An alternative here would be to use the summit, which would be a good idea too. And then we're going to fix with 100 base pairs around that center, so 50 base pairs either side. Now with that, we can use GitSeq. We can give it the BS genome object for our appropriate genome. I didn't need to extract the sequence first. I can just use the BS genome object directly. Can give it our newly centered peaks, so they're all going to be the same length, 100. And then I'm going to provide a name to this. Remember, we like to have names because if we ever write this to a FASTA file, the name we assign here will be in the ID line for every config in the FASTA. And finally, we have our DNA string set, 36,000 long. The names will be our peaks. Right? Is that the right number? 36,320 long. Yep. The names will be our peaks. Right? And then this is the actual sequence underneath our peak. Okay. There's something kind of cool there, actually. I like this. So this is the opposite. I don't think I've shown you this. This is the opposite to what we did with G ranges. Maybe I did show you this, but I still think it's cool. Ranges. So if we create a G ranges 1, 2, I ranges, sorry. Oh, it didn't like that, did it? No. G ranges, shouldn't. G ranges, seq names, chromosome 1. Ranges, oh, I ranges, sorry, I'm an idiot. What am I doing wrong there? So string equals thing. Oh well, I can't quite work out what I'm doing wrong. I'll have to come back to that. Oh, could not find function validate. Ooh, that sounds like something internal and horrible. I'll come back to that in a sec. But effectively, what I was trying to show you is that you can go the opposite from a character. So we could turn chromosome 1 into a G ranges. Does that work? Maybe. Let's try that. Maybe that would save me. No, I think I've done something wrong to my G ranges. We can turn a character into a G ranges. We can also turn a G ranges back into a character. So we can do as.character and then just turn our G ranges into our names. Right? So that's a nice little trick here. Okay. So now we have our sequences here. So this is what we want to scan. We have our motifs we want to scan with, right? So I think we got these over earlier on. Now we can use these together. And we're going to use match motifs. We're going to give it the motifs we want to scan, just the first 4. We'll give it 100 sequences that we want to scan through. And I'm here going to specify that we want to get the positions back. Right? So I want to know where in my sequences did this motif match. Okay. This gives me back a motif list, sorry, and then the length of this is going to be 4, right? 1 for every motif we've scanned. Okay. So if I look at my first element in my list, so I'm going to take now my motif positions here. This is my list. I'm going to subset it down to the motif I'm particularly interested in. And I can see inside there, I have this iRanges list for every single peak I've searched in. And you can actually see most of them are blank. Right? So 100 corresponds to these 100 sequences. Most of them are blank. It's just because we haven't found a motif in this peak. Right? So for this particular motif, it wasn't in these peaks. If we wanted to make this a little easier for us to read, we can actually just assign this here to another object. I'm going to give my peak seeks some names. So I'm going to give them some names. Again, the same trick we're doing. Just going to take these names from here, assign them to these list elements, right? Because they're all blank at the moment. And now when I unlist it, and I say use names equals true, it throws away all these blank elements and it just keeps the match I had, which is named from the actual peak name here. Okay. So I know that in this peak, starting at position 56 in the peak until position 69 in that peak, in that region sequence, this is where the motif was. Okay. So that's kind of useful. It means we can actually start to dig into our motif, into our peaks, and see whereabouts our motif, and hence probably our transcription factor, was within there. Often we may simply want to map motifs to their ATACseq peaks without really caring whereabouts in the peak it was. I just want to know true, false, this peak has this motif. Okay, so we can use that. We can specify that by changing none of our inputs apart from this last one. Out is now no longer position. It's going to be matches. Okay, so I think previously positions, now it's matches. Okay, when we run, we're going to use all the motifs and all the peaks here. Now when I run that, I'm going to get back a different class of object. Previously, I had this list, and every element of that list was a motif. And inside there, we had this iRanges list of where in each peak it was. This is going to give us back an object we actually hopefully recognize, a summarized experiment object. And summarized experiment objects hold matrices. And actually, it's going to be in the assay, the matrix for this summarized experiment. It's going to have our motif matches. So now we're not going to have samples. We're going to have 36,000 rows. Those rows will be our peaks. And now along columns, rather than the samples, we're going to have different motifs, right? So we're going to have, hopefully we can see it in the next slide. Brilliant. Okay, so this is our way now of storing this information. And the column names are our IDs. And the row names would be the peaks if we had row names. Okay. We can actually retrieve this matrix of matches, like I was saying, peaks by the motif, by either extracting it directly. We can do assay on the summarized experiment. We know enough about summarized experiment objects that we can interact with it directly. Motif matcher actually provides you with a function, motif matches, and you give it the object returned from this match motifs. And it's going to give you back a matrix. And this matrix we can then go and see. It's 36,000 rows, 746 columns. This is the matrix we wanted to get. And if we just look at the first 8, we can see it's a slightly strange matrix, but it still contains the same information as if it was a normal matrix. And what it has, because it's only true or false, this is what we call a sparse matrix. And where we would have a 0, we have a dot. And where we would have any value, we'll have this dash, vertical pipe. Okay, this allows us to save space. Okay, by having this, when we're going to have lots of zeros, right? We don't need, like with RLE compression, we don't want to have zeros inside our memory. We don't need to. We can save space by compressing this. And when we need to operate on this matrix, R will translate it into something we can read. This is it, right? But under the hood, R is going to handle this big matrix much better because it's a sparse matrix. Okay, so we can still use our matrix operations on this. Again, under the hood, it's going to know how to deal with a sparse matrix compared to a standard matrix. It's invisible to us. So if we use col sums on this matrix, knowing that the columns are our motif IDs, so these are the motif matches, and these are the peaks. If we do col sums, we can get an idea of the total occurrence of our motif in our peak set. So already I could say in our peak set, underneath in those sequences, underneath our peaks, we have very few of this motif, but quite a lot of these motifs. Okay, so we can already operate on this matrix just as if it was a standard matrix. We can also do things very simple here, like just get the peaks, right, which contain a hit for a selected motif. So I told you here it's dot and a pipe, but inside R it makes no difference. We can say 1 for a match, it would be a 0 for a not match. And in this case, I'm going to look in our matrix, this matrix here. I'm going to look in a particular motif. So I'm looking at a particular column. I'm going to say, just give me, for this motif, the amount of times when we had a 1. So give me the peaks which had a match for this motif. And just pull them out, standard indexing. And now I have peaks with this motif in them. Okay, and this might be something, I'm interested in only peaks bound by Mick. I can quickly pull them out this way and then do my further analysis. So I guess when these people, when Alicia Shep, who wrote Motif Matcher from the Greenleaf Lab, when she wrote that, she was writing it with in mind ChromeVar. Okay, so ChromeVar is a package which is going to allow us to summarize ATACseq signal not to peaks, right, but to be able to make a way of summarizing these up to the motifs. Okay, so all the peaks which had a particular motif, it will be able to make a meta peak, and it will look peaks with, say, Mick motifs. They tend to go up or they tend to go down across conditions. It's ChromeVar which really allows us to investigate this. So yes, as I said, with this summarization up to the motif level, we can start to see which motifs are important in some samples visually, but also we can do it statistically too. To load it, we just do library, ChromeVar. We can Google it to make sure we know how to install it. Oh, it's not the top hit. It's interesting. Copy and paste, and we're ready to go. So to identify motifs for ChromeVar, we will use Motif Matcher. They're written hand in hand to make sure they work well together. So we can provide our range summarized experiment object. This contained our counts over our peaks. And we're going to provide that directly to the match motifs function. But first, in order to make my life a little easier, we're just going to remove any peaks with less than 5 reads. Okay. Motif Matcher does something like this internally. I think it has its own function to allow you to remove low count peaks. In this case, we're just going to look across all the counts we made in our session before. Just make sure that in a row, there's at least 5 reads in any samples. Okay, so that should get rid of just really tiny peaks, which are useless. Now we got our filtered range summarized experiment object. We now want to correct for any potential GC bias. If you remember when we were doing some of our FastQ quality treks, generally there is a bit of a bias in how well GC is sequenced, GC rich regions are sequenced. So we want to account for this. So it's going to look across different sets of peaks and try and work out from their composition, is there a bias? So to do that, for us, we just need to run add GC bias from the ChromeVar package. We give it our range summarized experiment object, right? So our counts from our previous session, and we need to tell it the genome, right? Because it has no sequence information in our range summarized experiment object, but it does have locations, it has the ranges. So if we give this our counts and the BS genome, it's going to use the row ranges from our range summarized experiment, look up the positions in the genome, and then do this GC bias correction for us. Having corrected for our bias, we can then use this match motifs function again to identify motifs under our ATACseq peaks. Rather than supplying just the sequences here, though, we're going to supply the motifs we're going to scan. So this is the same scanning we were doing, same motifs we were using for just match motifs earlier on. We're going to give it our counts, right? So our range summarized experiment, no sequence data, but it knows where the peaks are. And then if we give it the genome as well as a BS genome object, it's going to know, okay, these are the ranges, and I will look up these ranges in this genome, and I will be able to then scan for motifs in those sequences. So it's doing a lot of work for us, right? It's actually extracting the sequence from this object by looking at the row ranges in our summarized experiment object. Okay, so then we now have our range summarized experiment object. We still have 36,000 peaks. Okay, so we had that before, right? Those were our ranges, but now rather than having counts in our object like here, we've now got the assay containing our matches, and actually we can see here the column names for our motifs. So now our rows are our peaks, and our columns are our motifs. So following this identification of our motifs and our peaks, we then can perform this summarization step using two functions, this compute deviations and compute variability. Okay, so we're going to run compute deviations on my counts. We're going to give it the annotations here, motif X, right? So these matches we found here, we give it our counts, we give it the result from here. And then with this, we then compute the variability across our data as well. So the deviations are computing how much, say, peaks with MIC differ from the average peak across samples. And then we can compute the variability of this. And this gives us an object we can start to do some analysis with there. Useful thing we can extract straight away is this deviations. From these things, we can actually get the scores here from these deviations. So here we can do deviation scores. We'll give it the deviations, and then it's going to give us back this dev Z scores. And it's telling us how much a motif is upregulated in that particular sample. And it's by sample here, right? So in this case, we can see that, peaks, ATACseq signal peaks with this first motif appear to be slightly up in this sample, slightly up in this sample, slightly up in that sample, up, but then down in these samples. Right. Okay, so we can now get an idea in this matrix returned from here. We have samples along here, and we have rows, whether or not peaks associated to this motif are higher or lower expressed in that sample, like higher, more or less accessible in that sample. Okay. So we can actually have a look at the variability result from this, and this will rank motifs by those which show the highest variability in the Z score. Okay, so here we get the variability. I think I returned this here, variability known. If I order that, we'll then get at the very top here, GATA, showing that GATA peaks, GATA motifs, or ATACseq peaks with GATA motifs are changing the most across our data. Okay, so then we would start to think that GATA is probably very important in the differences between hindbrain, liver, and kidney. Right? SOX42, tau, and this. Yeah. But this is what we would interpret this as. We can use the results from our Z-score deviations to identify in which samples our motif was enriched. Okay, so we could actually take this top 20 from our table here, this GATA, all these motifs which we believe are changing most across our samples. We don't know where they're changing. And we can just merge our most variable here with our table of dev scores. And we'll get a nice useful table where we can start to, in this case, we could say GATA. Now we have an idea actually what's going on. It seems to be down in hindbrain, down in kidney, but really up in liver. Okay, so we've ranked GATA by using this variability function here, compute variability, and it's told us what is most changing across our data, but we don't know where. When we combine that with the actual scores, these deviations, we start to get an idea per motif where this sample is particularly enriched. Sorry, where this motif is particularly enriched. Okay, really nice thing we can do with that finally here for this Chrome var bit is we can actually then take these deviations. So here, dev top. And I'm just going to take out the first two columns, right? Because they're going to be row name and name. They're not numeric. And then going to assign the row names to it. So I have some name here, which I can assign to my row names. And then I'm just going to make a quick heat map. No options, no configuration, just p heat map of our matrix. And this already gives me a nice quick overview about what is happening in my data. GATA is up in liver, but the RFX, the SOX, all these seem to be higher in the kidney and the hindbrain. There's also a little bit of variation there as well. Okay. So we can use this motif matcher to identify our motifs from Jasper. We can use then Chrome var to summarize peaks by samples into motifs by samples by saying whether or not all the ATACseq peaks which had a motif in them are higher or lower in that particular sample than the mean. And then we can visualize this to give us an idea. Okay, liver is enriched for GATA. These are depleted for GATA, but enriched for SOX and RFX. Okay. So that's how we can work with known motifs. And a lot of the time that is good enough. We take known motifs, we scan for their, we take known motifs, we have the transcription factor names associated, we scan for them, and then we can just go and start investigating, okay, this transcription factor, this motif, we can go and saying, well, maybe we knock this out or we overexpress it. It's going to affect our ATACseq data, and we can start to investigate these, whether or not it's really controlling these open regions. But this assumes that you know what you're looking for, really, and it assumes that you are interested in known transcription factors. I think there's definitely a move to also start to account for things like mechano transcription factors, things which aren't quite so well known. Right, we're not going to pick up anything novel if it's well, we're not going to pick up anything novel using databases. Right, we already know that motif, and we've already have a gene associated to it. If we want to look for new motifs in our data, so new patterns underneath our binding events, underneath our open regions, we're going to have to use something like de novo motif discovery. Okay, so we did this a little bit in our ChipSeq. We took the sequences under our peaks and we fed them to MemeChip. MemeChip, MemChip can do the de novo motif identification. And then once it finds this motif, it can try and associate it to the known motif it looks most like. Right, so it starts de novo and tries to work out what this motif may be. So here we're going to use MemeChip in discriminative mode to identify de novo motifs enriched in one sample over another. Okay, so I think before we were just giving in a set of peaks. Now we're going to say, I want to know what is enriched, what motifs are enriched in this peak versus this set of peaks. So the first thing we need to do is identify a set of differential peaks. We already did this in our last session. So we can just take DSeq, we load again these counts. These are the counts we just did motif match R and chrome var on. And we're just going to add in the factor information so we can get our data frame. And this data frame just has group. Right, so we're going to test over this. I give DSeq our counts, and I'm going to tell it this is our summarized experiment object. And I'm going to give it the design. I'm telling it it's by group. And then we give it to the workhorse function. It will go do all our analysis. As with all DSeq data sets, we then want to run results. We're going to give it our DSeq data set. We're going to tell it the contrast we're interested in. Within group, I want to have liver over kidney. It's going to be liver minus kidney. But compared to our standard RNA-seq, I want a G-ranges back because G-ranges allow me to do all these other operations. Right? I can use G-ranges to extract sequence. I can use G-ranges to annotate in ChIP-seeker. I'm then going to order this by p-adjusted. And then I'm just going to filter up regions, those which were greater than 1,000. Sorry, the first ones which are up, right? The first 1,000 which were up, the first 1,000 which were down, right? Ranked by our p-adjusted p-value. And we can just look here. I haven't got the p-adjusted, but these would be my top 1,000 up in liver, and these would be my top 1,000 down in liver, up in kidney. For MemeChip, we want to resize the regions to 100 base pairs. This is because by default, MemeChip will do the trimming for you if you don't. So just to make sure we're giving it what it's going to operate on. So we can resize our regions. We'll fix the center, make them, again, 100 base pairs around that. So it's quite a small region. And we'll do that for the up and the down. And then we'll use our getSeq function. And we'll get the sequence for our G-ranges from the appropriate BS genome object. Do that twice. I'm going to turn my G-ranges. These are my G-ranges here. I can turn this into a character by doing as.character. Right? That gives me a good name to assign to my DNA string set. I can do that for the down, and then I'll write that out to upregions.far, downregions.far. This is our FASTA file containing all the sequences under my peaks which went up in liver or which went down in liver. So then we would go to our online submission portal, and you can have a look here. I only really wanted to bring you attention to this part, discriminative mode. And this just means you're going to upload rather than one FASTA. Might upload. I don't think I've got the data here, but you would upload two FASTAs, one of your sequence here, and then your control sequence here, and it will go and run the analysis for you. Okay, I think we have the data actually sitting in our directory, so we can go have a look what the result was like. Data, MemeChip result. Okay, so then, oh yeah, look, great. So unsurprisingly, GATA came out as our top hit. Not centrally differentially enriched, right? And they're doing this because we haven't bothered trying to get the summits, but we don't need to for this. But, you know, by very quickly, we've managed to do the same thing we found in the scanning with chrome var and motif matcher. We take the sequences of those which we knew were up in liver and those which are down in another condition. And again, we're managing to pull out GATA as our top hit. And this is the actual motif. But as you can see, it associates it to motifs it kind of knows. And this is, I guess, what they consider the GATA motif. See it matching. Okay, so that's nice that we ended up with getting the same result back by a totally different method. Right? So we can either take the regions which are most up, most down, give them to meme chip as a comparison. And it's going to say in liver, I tend to see GATA in those which most go up. But in this method we did here, we had no cutoffs, right? We just summarized the peaks to GATA and we still see the same thing. So totally different methods of showing us the same results, which is very reassuring. Okay, so I said at the beginning, you're not stuck though with transcription factors which are pre-built in these databases for you. You may go and discover a novel transcription factor, and certainly it's happening at Rockefeller very regularly. So you still want to use Bioconductor software. We still want to integrate our data with ChromeVar and known motifs, maybe plot them side by side. We have luckily a really cool package, Universal Motif. Again, I think it's Bioconductor. Yep, we can just copy and paste if we want to install. So this library is really the nice kind of connector between all these different motif packages and external formats. Although TFBS tools allows you to operate on these well, it is a bit of a pain. If you ran meme chip, you get results back in meme format. If I ran Homer, I get results back in Homer format. I need some way of bringing these into a consistent object so that I can use them all in ChromeVar. And this is what Universal Motif is. So obviously a really relevant function for us is read meme. So if we went into our data, when we downloaded it, we got a file called combine.meme. Read meme will just read this straight in. You can see here, here's the first motif, actually identified by DREM, which is another program. It's called it 1. It gives you some information here. It tells us the consensus. So I guess you guys can get a hint why it's called GATA. And if we want to, then we can start to convert this into something that we really want to have, which is a TFBS tools PWM matrix. We just need to use their additional function, convert motifs, meme motifs. Right, so this is what we've read in here. We're going to convert this to a TFBM's PWM matrix class. And now we have these objects which are the same as what we would get from TFBS tools. And then we could do all our plotting, visualization, and the same thing. So we can go back from our meme chip back into our world of TFBS tools and do all our scanning again in Motif Matcher if we wanted to. So we got to the end of our course for today. I've got a few exercises which I think were kind of cool. So perform a differential ATACseq on the kidney sample versus the brain sample and write out the FASTA of the top differential. 1,000 ATACseq peaks up and down for kidney minus brain. Great. Extract the sequences from these and submit to meme chip. Okay, so we're going to do just what we did for the liver versus kidney. We're now going to do kidney versus brain. And then again, that's one way to do it using meme chip. Take the top differentials and then do a comparative motif analysis. The other way to do it would be ChromeVar. And I recommend you step through that as well and see how similar or different these results are. Again, all our answers are here. I think we had a link somewhere to the actual results from meme chip as well. But feel free to run through. I will be in the room until 4:30, and then I've got to head off. If you have any questions, you can either raise them here on our issues page or in a further session with Matt. But for now, I'm just going to hang around. And I'll be around if you have any questions on the exercises. I didn't actually see any questions coming. All right, thank you very much for the day. I'll speak to you all. If you have questions, I'm here. Otherwise, I won't see you again in these courses, but I'm sure I'll see you all somewhere in the future. All right, take care, guys. Bye now.